Madam Dr. Chandrima Saha, President INSA and Fellows of INSA, speakers of today's symposium from Lucknow and also IIT Kanpur, my colleagues and attendees. So on behalf of CDRI, it gives me great pleasure to welcome you all to this interesting symposium organized at CDRI by the INSA Lucknow chapter. All of us understand that importance of basic sciences, not only to understand fundamental principles of life and also physical world, but also its essential role in leading us to sustainable progress. So in order to recognize this, the United Nations General Assembly declared this year, that is 2022, as a year of basic sciences for sustainable development. We know that in our CSR system, we always talk about translational science. Of course, without basic sciences, we all know that we cannot be successful in translation science. So that's why basic sciences is very, very important for all of us. So today's symposium covers many aspects of this broad theme. And I'm particularly happy that in addition to researchers in biology, we have academics from physics and also electrical engineering speaking on interesting topics in today's symposium. I was also told that there is a session with the research scholars that covers diverse aspects of the role of basic research in the achievement of SDGs. So SDGs are Sustainable Development Goals. So I wish you all the best, and I'm sure you all going to have scientifically enriching day. I wish you all the best. Dr. Saman.
Thank you, Dr. Reddy, for these remarks. We're really honored to have Dr. Chandrima Saha, President of the INSA, joining us online today. Dr. Saha, please.
Yes, Dr. Reddy, INSA fellows, my colleagues and friends, it is indeed a happy occasion to be here. And I'm pleased that the Lucknow chapter took an active interest to organize this mini symposium under the International Year of Basic Sciences for Sustainable Development. The celebration of the International Year of Basic Sciences is an activity that has been taken up all over the world, and the program is based on 5 themes identified by UNESCO. The program aims to narrow the gap between society and science and technology. We know that science and technology provides the power to achieve several developmental goals, which should be possible without compromising the environment. And they include strengthening the presence and visibility of women, basic sciences as a source of international dialogue and peace, science as a global public good, innovation and economic development, education and human development, meeting broader global challenges. Development is every society's dream, in fact, and ever since humans appeared on Earth, the process has been on at various phases for millions of years. And no doubt that the material developments that we have made over the years has made our lives comfortable and elevated our standard of living. But alongside this process, we have plundered the Earth so brutally that we have been forced to wonder, have we really progressed? The footprints left during the ongoing Anthropocene does not augur very well for us. We cannot hope to sustain in the long run at the present rate of exploitation of resources and ruining the environment. In the year 2015, the United Nations General Assembly adopted Agenda 2030, an integrated vision for sustainable development for all the world's populations. It is articulated into 17 sustainable development goals towards which the whole humankind has to collectively act. It starts from no poverty, zero hunger, good health, quality education, gender equality, clean water, decent work and economic growth, reduced inequalities, life below water, life above water, climate action, justice, and to get these into action, we need partnerships for the goals. So that is the most important issue. So these sustainable goals, all of them require input from science and technology. And the debate on relative importance of basic versus applied science is not new. We have discussed this over and over. But even when technology has grown independent of basic sciences, its efficient utilization becomes possible only through the understanding of basic science behind it. Last 2 years have taught us that basic science is never irrelevant. How could one think of finding medicine or vaccine for SARS-CoV-2 if the basic nature and properties of the virus were not known through basic sciences? If basic science did not fuel the advent of modern technologies related to diagnostics, treatment, and prevention. The topic chosen for today's presentations are the topics that are chosen for today's presentations are very comprehensive. I think they're very contemporary, they're forward-looking and relevant. I'm actually looking forward to these deliberations. And for this, I congratulate CDRI Director, Dr. Srinivasa Reddy, and the Lucknow chapter convener, Dr. Saman Habib, to be the first to block and organize this symposium. We still have time for the IYBSSD activities that will culminate with a ceremony during September 20 to 22. Other chapters should take cue and propose activities for under this UNESCO action. Any group can propose an activity to celebrate this international year up to 30th June 2023. I thank you all for making this event possible, and I'm sure that an intellectual feast awaits you. Thank you very much.
Thank you very much, Dr. Saha, for putting across the main purview of the year for basic sciences for sustainable development across to the audience. We hope to have a constructive session today also to highlight some issues that are not directly related to science as we practice it in the lab, but the ramifications of scientific practice as we see in society. And for this afternoon, therefore, we have a very special session with research scholars who will put across their views on various aspects that are covered by this theme. The first session today is a scientific session with two very eminent speakers. I would now request Dr. P.K. Seth to please come up on the stage and to chair this session. Dr. Seth is the chairperson for this session. This session has two speakers, Dr. Vandana Prasad, who's Director of BSIP, the Birbal Sahni Institute for Paleosciences, and Dr. P.K. Singh from the National Botanical Research Institute.
Thank you very much for this opportunity which I have to chair this, the first session which is being organized on the International Year of Sustainable Development, role of the basic sciences. I think we are starting a couple of minutes earlier. I do not know if there are people are timely to want to hear the lectures. But anyway, first of all, I'd like to congratulate CDRI Director Dr. Reddy and Dr. Saman Habib, the convener of this Lucknow chapter, for taking up this very, very important activity. And as our president mentioned, is the first one, Lucknow chapter, to start this activity. Very well said by Dr. Chandrima Saha. The role of the basic sciences. I think I look at basic sciences are involved in almost all the 17 goals which have been set up by the UNESCO. And what is important is this, that we know it is the good science which drives the technology, whether it is the development of the technology or its translation of the technology. It's the basic science which is very, very important. And I think this is once again very important issue, that how science is relevant to the society which is being taken care. So I have great pleasure in introducing the first speaker. We really have two distinguished speakers presenting their basic findings. And Dr. Bandana Prasad is the director of the Birbal Sahni Institute of Paleosciences, which is in Lucknow, is known as BSIP, as research interest in is a large of research interest which includes pre and post condition biotic collusion of the biotic turnovers and climate changes pertaining to the India particularly and the plant biodiversity, early evolution of the grasses and grazers, earth warming events and climate change. She has been recipient of several international fellowships and academies awards, and she is a fellow of the Indian Academy of Sciences, which is located at Bangalore. She has chosen to speak on evolutionary history of angiosperms, evidence from fossil recorders. Dr. Vandana Prasad, the floor is open to you now.
Dr. Chandrama Saha, President INSA, and Dr. Reddy, Dr. Saman Habib, Dr. Madhu Dikshit, Dr. P.K. Seth, Dr. Kamboj, Dr. C.M. Nautiyal, and many more, all ladies and gentlemen. It's a pleasure to be here to deliver a lecture for this, this particular symposium. So my topic is, please, can you give the slides? So my topic is evolutionary history of angiosperms, evidence from fossil records. So Birbal Sahni Institute of, let me talk about the institute, the Birbal Sahni Institute of Paleosciences. It was, it's an autonomous institution in the Department of Science and Technology. This institute was established in 1946 by Professor Birbal Sahni to carry out paleobotanical studies. Recently, the institute has been changed from paleobotany to paleosciences. So now the scientists of this institute are working on several aspects, right from climate change, evolutionary history, and other contemporary issues. So talking about the topic, the angiosperms, the evolutionary history of angiosperms, we all know that angiosperms are the most species-rich and ecologically diverse amongst the plant ecosystem. We also know them as a flowering plants. So there are some special morphological and ecophysiological traits which make them succeed over any other type of plant system, like presence of carpels, flower structure, bisexual character, double fertilization, cross-pollination mechanism, nectar production and pollination through different agents, fruits and seed dispersal. And the ecophysiological traits are xylem vessels for effective water transport, leaf venation, and stomata. So there are several studies has been done on these aspects, and so scientists, they have of the opinion that when the angiosperm evolve, the climate also becomes very humid, because they only in angiosperm they have the capacity to transpire so much. So because of in tropical area, we all know that the water vapor which comes out from the after the transpiration from the plants, they provide rain clouds. So when we see the deep time geological history, we see that angiosperm, when they came, the climate also become very humid. So apart from that, the angiosperms, they have stomata. So stomata, as we all know, they help in gaseous exchange. So angiosperm, they have the capability to modify the number of stomata. If there is a high content of CO2 in the atmosphere, the plant will adapt. It will modify the stomata will be reduced. And if the CO2 level is very low, then the stomatal frequency will increase. This we have seen in the past also, and for last 100 years or 200 years of data. So when did the angiosperm evolve? So records the study So the study says that the angiosperm evolved during late Triassic time period at 230 million year time ago, and that time that most of the northern continents, they were in the tropical region, and there are few records that can tell us that the angiosperm, they arrived at that time. Later on, then there is a Jurassic period that was a big period, 184 million year time span. But this time we don't see any angiosperm record, probably because this time period has been identified as a most arid period in the geological history. Or because of the aridity was desert areas were so much that angiosperm records cannot be seen. So next comes the Cretaceous time period. During the Cretaceous time period, there are several records and angiosperm, they started coming, and then later on the angiosperm records showed highly diversified conditions, and they were proliferating in each and every part of the globe. So angiosperm, as compared to other plant groups, angiosperm came very late as compared to gymnosperm fauns, but when they came, they completely diversified, so much proliferated and covered the globe. So when we talk about the history, we see that apart from geological history, the Indian continent has a very dramatic history. As you see, this is how that the continents where they were moving, and you see that Indian continent was in the southern high latitude, and it started moving during early Cretaceous time period. It moved further north. It crossed the equator and then collided with the Asian plate, and that is how the mighty Himalayas originated. And so in India now it is at 21 degree north. So you can see that it travels from 60 degrees south to 21 degree north. So it has a the biota which was present on Indian subcontinent during different time span. It crossed several latitudinal changes. So these latitudinal changes must have given a very important criteria and important insight to understand how the evolution has taken place on Indian subcontinent. So we see that during, as I said, that during Triassic time period, there were a lot of desert areas were there. So there are less records of angiosperm, 1 or 2. And in Jurassic time period also, the desert areas were very high. So we don't see any records. But during the Cretaceous time period, when it started, there are a number of angiosperm records started appearing. And you can see the desert areas, they were very few. So this has actually given impetus to the evolution of angiosperm. And you also see that the continents where they started drifting from each other. At Cenomanian time, all the continents, they were just like an island. India was at the mid-latitudinal position, and there are a number of angiosperm records you can see all over the globe. And during all these periods, there was no ice caps in the polar region. And Maastrichtian time, when there are so many records of angiosperm, these angiosperms, they show their similarity with the living angiosperm population. So early angiosperm diversification took place in Gondwana. So higher CO2 level was there, equable climate, because there were no ice cap on both the polar area. So the polar area climate of and the equator climate, they were almost the same. The lack of polar ice sheet during early Cretaceous and early Paleogene led to the origin of tropical angiosperm. So why was Cretaceous and early Paleogene so warm, a period of sustained greenhouse Earth? So if you see both the Paleozoic, Mesozoic and Cenozoic era, you see there are a lot of volcanic activity that took place during Mesozoic time period, and also during Cretaceous, the volcanic activity was so much because the continents, they were rifting from each other. This has resulted in a lot of volcanic activity. In Cenozoic time period also, there was a lot of volcanic activity. But apart from volcanic activity, there was one particular time period that is known at 55.5 million year, Paleocene-Eocene boundary interval. That was a period of 200,000 years of interval. That was extremely warm, is considered as a super house greenhouse period. And because this was the time when the CO2 level was 6 to 8 times higher than the present day value. And because this was because there was a massive rupture of methane hydrate from the ocean bottom. So actually this particular PTM event is now being considered as an analog for our future climate also. So next please. Thank you. So when we talk about the evolutionary history, so one particular term, that is paleobiography. Paleobiography is very important. That is the study of distribution of evolution and radiation of organism through space and time, study of evolutionary relationship amongst the different organisms, inferring age and place of origin of organism, assessing the role of post tectonic and climatic events in the evolution of organisms. So there are three fundamental processes are required in paleobiography: evolution and speciation of biota, both plant and animal, extinction, and its dispersal. So we need fossil records, we need dated phylogenetic tree, we need the current distribution of biota so that we can develop a paleobiographic history. So for developing the paleobiographic history, there are a number of methods. So one particular method, that is a phylogenetic analysis, which is extremely important. That involves the study of present day biota. So like these species are A, B, C, D. It's moving. I'm so sorry. So there are a number of species like A, B, C, D. These are the living species, and their character that make them clade 1 and clade 2. So their divergence ages can be determined, and this could be their divergence age. So similarly, we can also we can also decipher the root of these particular family or clade. So this is particular phylogenetic study is extremely important in deciphering the paleobiography of biota. So predicting speciation, extinction, and impact of climate change on diversification of flora and fauna becomes easy. And this holistic approach of integrating multiple disciplines ranging from paleobiology to molecular biology would address the issues of impact of climate change on species distribution and their habitats, phylogenetic relationships of extinct and extant lineages, predicting speciation and extinction in response to historical processes, and finally we can develop conservation measures of a species by taking into account of evolutionary and ecological aspects. So we have the DNA data of living species, we have its fossil records, we can develop a phylogenetic tree, we can predict the speciation and extinction of flora and fauna in response to past geological events, and also predict the impact of past climate on diversification of flora and fauna. Let me take you to Rajasthan Desert, Rajasthan area. It's a Thar desert. There are a number of lignite mines are present in which we are working on and trying to trace the paleobiodiversity in the deep time. So in these lignite mines, we get a lot of fossils. These are the insects, these are the vertebrate fossils, these are the pollen. Apart from that, there are many megaflora also present in them. So when we try to see their similarity with the living population, we find that they are like this type. Lemurs were present in India at one time, but now they are no longer present in India. They are found in Madagascar only. They are extinct from India. There are tapirs, they are found in South America, but now they are not present in India. They are found in somewhere else. Like many plants, they were present in India at one time, but right now, because of the climate change, they are no longer present in India. They are residing somewhere else. So this is how that we can predict also that how the climate change is taking shape. These are some flora which are which is found in only Madagascar, but in India we were getting at one time. So this work we have done in 2009. We have found that 55 million year flora which was found in India, that shows its similarity with the flora which is found in Western Ghat area. So this is the area. This is the area. This is from 9° to 10° latitude. This particular area has an endemic population. Most of the plant and animal species are monotypic and endemic. So if we consider the mean annual rainfall condition, then this area becomes very, very wet area. And if we consider the lesser number of dry months, then this area becomes very wet area. So most of our fossil records are found from this area. So we come into some case histories. So we have done some evolutionary history of Abidaceae. Abidaceae family is a pantropical family and which is used for timber and fruits. The family consists of 500 species. So we found its fossil records in Rajasthan mine and Gujarat mine. So these are the fossil records, which pollen fossil records, which we have found, and they have the similarity with the living population of this family. So we have done the morphological data. 20 characters of pollen have been considered to develop a phylogenetic tree. This is the character matrix using the morphological data. The lesson 0 and 1 has been given to the primitive character, and the more advanced character has been given a higher number. So we have also taken the molecular data of this family of living species, and we developed a tree. So this is the tree which we have developed. This is the phylogenetic tree, and then what are the characters, how the characters evolve in this family. And this family originated around 106 million years in Africa. And from Africa, it was transferred to India. And later on, it was transferred to Southeast Asia. Another history is evolutionary history of Dipterocarpaceae. Dipterocarpaceae family is a timber producing, we all know that is a sal family. And it is found mainly in Southeast Asia. Southeast Asian rainforest, about 80% of population belongs to this family. So if we see that this family is how it is distributed, and this family show disjunct distribution in Gondwana continents and Southeast Asia. So there is one genus found in South America, 2 in Africa, 1 in Madagascar, 1 in Seychelles, about 7 in India, in Western Ghat area, and about 10 in Sri Lanka, and maximum in Southeast Asia. So that shows that this family must have originated in Southeast Asia. But we found the earliest record during Cretaceous time period, we found its records in India and Africa. So that suggests that how this family in ancient time, in deep time, was present in Africa and both in India. We did the similar exercise in similar methods. We found all its genus in India at one time during Paleogene time interval, 60 to 55 million year time span. So these are the fossil records of this family. And we did the dated phylogenetic tree has been created. The family originated in seasonal climate of Africa around 102 million years. Family dispersed from Africa to India via Kohistan Ladakh arc around 70 million years. So how these clades have been identified, and they show their divergence from one another. This has been done recently. So this is when India was moving towards north. At that time, first it made in contact with Kohistan Ladakh arc plateau. So these are the small island chains which were connected from at one end at Kohistan Ladakh arc and one end towards Africa. So when India was moving towards north, first it made contact with these island chains, because India and Africa, they were never together after they separated during deep time Gondwana time. So no question of coming close to each other. So through these island arcs, majority of the tropical population that was thriving in Africa, because Africa was already in tropical zone and India was moving towards the tropical zone. And so from Africa, through these island chains, these tropical angiosperm, they were transferred to India. And when India further moved towards north, these island chains, they were subducted or consumed beneath the Indo-Asia collision zone. So this work was in January, was published in Science. apart from that, there is another history that is a family is called Mauritania. That is a family of subfamily of Ericaceae. This is a palm family, and we found their fossil records in India during Paleogene time interval, Paleocene time interval, and presently it is no longer present in India. It was present in South America. And so, how this family diversify and how they got extinct in India, and later on they diversify in South America. So this is all this data that tells us that many biota, both plant and animal, maybe they are present right now, they are present in somewhere else, but their evolution must have taken at different place. So this is like Southeast Asia, like about 80% of rainforest, which is dominated by the Dipterocarpaceae family. So it was, and there is a lot of research is going on, because this family is now endangered because a lot of cultivation and because human activity is actually they are removing most of this progeny and all that. So later on, how this family will behave, the past data and the past information can tell us that how this family will behave in future. So this is about that how this Mauritania was present in different continents, and so this particular family, this Mauritania palms, they are extinct in India before India made collision with Asia. And that is why this particular family could not be transferred to Southeast Asia. So all this data that tell us that the biogeographic history of both plant and animal is extremely important when we are facing threats like global warming. So there are many species of plants like Chironji you see. It is going to extinct in future. There are many plants like because they are not being able to withstand the changing climatic condition and high CO2 level. So we have also found earliest records of grasses in India, and it was published in 2005. Then we have also found that wild rice variety, which was present in India at 65 million years ago. And because there is a lot of hue and cry between India and China that rice came first in China or whether it came first in India. But if you see that already the stock and the genetic stock of rice was present in India before the collision with India and Asia. So probably from India it has gone to China. So I think so these are our lab people who are working on plant biogeography. So we have expertise in our institute, we have expertise in different areas of evolution, morphology, taxonomy, high resolution biostatistics, which is useful in hydrocarbon exploration. Then again, the paleobiography, paleoclimate, paleoecology, paleoenvironment, paleomagnetism, environmental magnetism, paleontology, vertebrate and invertebrate paleontology. We also provide geochemistry techniques, geochronology, radiocarbon, TLOSL dating, elemental inorganic stable isotope geochemistry, organic geochemistry, petrology, archaeobotany, dendrochronology, industrial palynology, and ancient DNA. We provide consultancy services in these areas. Thank you so much. I would be very happy to answer a question if there are any.
Thank you, Dr. Prasad, for that talk. I just had one question. Based on the work that we do in terms of how, say, angiosperms have evolved and how they've migrated over ecological evolutionary time, with the current rapid climate change, what are the predictions that one may make for the future of angiosperm diversity in India and also in South Asia in general? So is there a way of looking at particular families of angiosperms, and what are these ways? I mean, how does one do that kind of prediction?
So first of all, it is extremely important to understand the plant physiology, the particular plant, how it is require, what kind of environmental conditions. And also the one particular area which is now developing in India, but is a climate, species distribution modeling. How the species will migrate from one place to another due to climate change. So for that you need a different time span history of that particular plant group or animal group. So like 3 times, like one Holocene, then about 5,000 year, what was the condition of this particular group has been distributed. By then doing the lot of statistical analysis, then we can develop a model. Like I told you that the beauty of that particular, like जिसको आप लोग पलाश कहते हैं, so that particular plant is now thriving in central India. But if there is the drought periods are more, so this particular plant is going to shift towards a more moisture condition where there is a not seasonal variation, much seasonal variation. Like April has become very hot and arid. So many of the saplings, like the plant, they regenerate during this time period. So what we see is that there is the picture is emerging. It's not very good for the country, and we need to do some conservation policies has to be made for conserving these plants and animals. Yes, please.
A very nice talk, ma'am. Just a hypothetical question. Yes, it can be scientific at the same time. What was so great about South Africa when we learn about evolution, origin and evolution, say with respect to these plants as well as humans? Originated there and then spread over. So origin and evolution ultimately goes back there. And was there, I mean, some co-evolution and co-origin? And ultimately, again, connecting to ma'am's question, how have we affected the evolution of these plants today or till now?
Yes, actually, it's a very valid question. And because whatever, like we are doing basic sciences, but also we want to, like, there are so many lessons. Like when we talk about the global warming and how the global warming is taking shape and how the plant and animals, they are going to be affected. So it is extremely important to study the deep time also, because whatever like last 200 years or 100-year data cannot tell us about that what will happen in future, because the pace is very high. When I'm talking about the past global climate and deep time climate change, that took place millions of years. But now the thing is the pace of climate change is becoming very, very fast. And then the CO2 level, the anthropogenic emission and all that. So this is also now creating that. So it becomes very difficult. But if we understand the behavior of particular plant and animal, how it has behaved, how a particular family having about 64 species and about 20 species of, for example, 20 species died because of change in climatic condition in the past because of natural reason. So that gives that particular family is susceptible to change, or it is going to extinct, and it is very sensitive, or particular family which is very hardy. Like I said, there are certain plant community which are very hardy, and there is not much change. There is not much will be affected due to climate change. So all this data, when we gather all this data, then we can develop a model and can predict like that. We talk only about precipitation and temperature. Temperature is rising, precipitation is taking place like this, like this, like this, like this. But we never talk about that how the species will behave, how the species will be affected. We only concerned about human being, but we are not considering about that how the animals will be affected. Like particular plant is affected by is pollinated by certain insects or animals. If these insects and animals, if they die, if they are not no longer they are there, then that particular plant species will also die. So it is very important, it's very sensitive to understand the climate change in terms of that how the species will be affected. It's not a question of like something like, but it will take thousands years or something like, but it's very valid and it's extremely sensitive. Thank you. Yes.
India is dominated by certain crops which are not native to India.
Yes.
For example, wheat, potato,
Majority.
tomato, you know, guava, for example. And all these things have happened in last 400-500 years.
Yes.
Right. So what is your view on such large level of cultivation in our country?
Yes, majority of the crops like they have been brought from somewhere else. But major issue is like a particular community is changing the scenario also. Like you are bringing some particular variety which is taking a lot of nutrient, which is not adapted for this particular area. So it's taking a lot of nutrient. It is not pollinated by certain insect. It is not seed dispersal is not being done by certain animals or insects. So this is also extremely important that later on that the situation will become like this. You have a completely different kind of scenario, and many of the indigenous forms may be vanish, or maybe changed, or maybe modified, maybe. So these are some issues like I can't tell you about because I'm not a expert in this area. But definitely, because when we see things together, we don't see things like separately, like one particular family. How it is like in particular type of climatic condition and scenario, taking nutrient, what kind of nutrient it is taking. So considering all this thing together, then we can develop a model and the picture will emerge. It will be more, I think, beneficial, and it would be very good for people who are doing conservation, who are making conservation policies. Yes. Yes. So when we have to understand the climate change, we have to understand also what are the areas which will be affected by climate change most. So the areas which are affected will be affected through climate change are Himalayas. Himalayas, the Himalayan population like plant and animal, they are shifting from their own place. You don't find them. There are many areas like because of warming, because of high temperature condition, and these plants are adapted to a very specific kind of climatic condition. They have a very specific niche. So they are going to shift. They are the most drastic effect will take place in Himalayan region. Second is our coastal areas. So coastal areas also very prone to disasters and all this, which is happening, sea level rise and all that, which is happening. And second area is the interior part, like these areas, like those which are interior central India, will become very hot, very hot. So like when we talk about, when you see like month of April, for last 5 years, it becomes so intense hot region. So whatever like the plants which are going to like which will like seeds will be given and the plants will appear. So that particular those plants will never appear. Yes. Thank you so much, sir.
इससे आगे बढ़ेगा।
Eight research articles and 8 patents. He is a fellow of the INSA and is going to speak on white fly tolerant cotton for North Indian farmers. Floor is yours.
Thank you, Dr. Seth, for introduction. I thank INSA, INSA Lucknow chapter, Dr. Saman Habib, for giving me this opportunity to share some of my research that we have been pursuing at National Botanical Research Institute. Can I have my slides? So, topic of my talk is white fly tolerant cotton for North Indian farmers. So, I just want to create a background so that everybody gets involved in the discussion and talk. So, who are the most abundant on our planet? Is insects. There are around 10, lakh species has been recorded. And so I do not know what is that. Yeah. So there is a global projection that if insect pests and diseases are not controlled, world is going to lose crop yield worth 540 billion US dollars. Is huge. And as with this recent data from FAO, around 40% global crop yield loss is due to pest only, and that happens every year. So to control pests, you use pesticides. India is not a great user of pesticides. We have a perception that we use pesticides in excess, but it is not true. So most of the India ranks, 10th in the scale, and most of the pesticide companies are pushing their products in India. They want to sell because they see India as a big market. So let us talk about crop-insect interaction. So we are dependent on around 300 crops globally. The number can vary. So, and then these crops are actually attacked by insects, both in the field, which is called pre-harvest condition, and also during the storage, that is called post-harvest condition. So human has, improved crops for its quality and quantity, but that improvement also, encouraged insects to start, feeding on these crops. So, our intervention actually invariably helped insect population. And it is remarkable ability of insects that they can eat, wood where the water content is just 5%, and they can eat a food where water content is 95%. So very high, adaptability. So let us talk about cotton. So why cotton? So cotton is the third most important crop in our country after rice and wheat. It is grown on 30 million acre of land, 30 million hectare of land, and cotton is sold in our country worth 1.1 lakh crore rupees. So it has a massive impact on our economy. So cotton is a very resourceful crop. So it is a fiber crop, it is an oil crop, and it is, a feed crop. So when cotton is produced, around 30% of the produce is fiber, around 28% of it is oil, and then rest is set cake meal, which is used for feeding animals, cattle. There the protein percentage is something like 45%. Very rich in proteins. So this situation actually makes, cotton a very good host for variety of insects. So around 30 insects, 61 insects have been reported, and 5 falls in the pest category. So, if you see try to understand the economy, we grow cotton in, 13 million acre of land, which comes around 3 crore acre. So in Punjab, a good farmer actually spends around 20,000 rupees per acre on cotton and produces cotton, around 20 quintal, 10 quintal per acre. He spends around 8,000 rupees for harvesting and then sells the produce at around 60,000 rupees. The cotton price is 60 rupees a kg. So, from per acre actually they get something like 32,000 rupees per acre per season. But if the crop is damaged by some reasons, farmer doesn't get anything. It is notable that India ranks 37th in world in cotton productivity. So our unit area production of cotton is very, very poor. It is something like 470 kg, per hectare, which is very, very low. Most of the African countries are better than India. So there is need to protect this crop, and so that the same amount of cotton can be grown in a smaller area, and the field becomes available for crop like, chickpea, pigeon pea, mustard, so that oil seed crops are produced in more, pulses are produced in more quantity. So we work on defense in cotton against the insect pest. And the one point which I want to mention, that 45% of the total pesticides used in our country is used only for cotton and rice. So cotton and rice are the true core crops which actually consumes huge amount of pesticide, And pesticides invariably, creates a lot of problem. It is a soil pollution, water pollution, health of farmers, and many other things. So now I am coming to my topic that, in India, cotton is damaged by a total of 8 different insect pests. So one insect, Helicoverpa armigera, is controlled by BT cotton. People have a perception that BT cotton is tolerant to all the insects. It is wrong. Monsanto never claimed, but we developed a perception that BT cotton is tolerant to so many insects. So they use Cry1 AC and Cry1, Cry2AB delta endotoxin genes cloned from Bacillus thuringiensis for insect defense. At National Botanical Research Institute, we are trying to control, we have transgenic cotton, which are tolerant to pink bollworm, Pectinophora gossypiella, Spodoptera letura. These 2 insects are controlled with a protein, namely Cry1EC. We are trying to control whitefly with 3 insecticidal genes. One is Tma12, second is MSC14 and its 34 kDa version. There is another protein, namely PNU08. We are trying to control aphid, which is another insect pest of cotton, mustards, and many other crops. We are trying to control this with another protein, namely DHI31. But today's talk is focused on whitefly and Tma12. So whitefly is distributed all over the world, and it hosts several crops of horticulture, vegetable, and ornamental importance other than cotton. And the cause of increase its population, it has become a very aggressive pest. The cause is increase in the humidity, greenhouse agriculture, high density plantation. You want to grow more plants in a small area to produce more, to get more yield. And because of this, the insects like whitefly has increased. Nitrogen rich soil, ability of rapid multiplication. It is estimated that this insect alone damages crop yield by a value around more than $1 billion US. So what is a whitefly? I'm in CDRI. I know you know mosquito and malaria more than whitefly and viral diseases. So I'm just trying to compare that, in case of mosquito, female survives on human blood. It causes irritation and it spreads disease like malaria, chikungunya, and dengue. Whitefly is also very similar for the plants. It irritates plants, feed on nutrition-rich sap, excretes a lot of honeydew. It takes a lot of food from the plant and excretes a lot of carbohydrates which are produced in the plants. So it is called honeydew, which is very sugar-rich. And then honeydew invites, growth of fungus on the plant, and it spreads plant viral diseases. So this slide shows, how, whitefly damages a crop and how it causes spread of the viral disease in crops. This photograph shows the cloud of whitefly in sub-Saharan Africa. So in 2015, you recall cotton crop in Punjab and a lot of crop in Pakistan got damaged because of this insect pest. So this slide shows the severity of whitefly on cotton. You do not see these many mosquitoes at one place, but in cotton, you get huge amount of whitefly, huge number of whitefly. So in Punjab, pesticides worth, $50 million US was sprayed, but crops had to be uprooted. There was no yield, and it caused, loss worth 47,000 crore rupees. Just in Punjab, damage happened in Haryana, Rajasthan. Damage also happened in Pakistan, where we do not have a record, but a lot of damage has happened. And in Punjab, around 17 farmers committed suicide covered, it became a major news in 2015. So whitefly cannot be controlled with the pesticide if they are colonized on a crop. Known insecticidal proteins are not efficacious to whitefly. The GM cotton tolerant to whitefly could not be developed. Plant explored diversity to address this problem. We explored, plant diversity to address this problem and discovered a few insecticidal proteins. So I'm very glad that I did PhD in Lucknow and I interacted with scientists and students of CDRI, and there I had learned a word, activity guided purification. So what actually we did, we got several plants, right? And we thought that probably one of these plants may have a protein which may be toxic to whitefly. So you need to appreciate one point, that whitefly is a crop insect. It doesn't, feed. It requires a very high temperature, high humidity, and it is a crop insect. So it, attacks a few, 200 plant species. But, these plants like ferns and mosses, they are not, infested by whitefly. So the idea was that how to bring, plant biodiversity and whitefly together to find out something interesting in plants which can control whitefly. What is the strategy? The strategy was you collect, leaves and root of fern and mosses on the whole. So leaf of the fern is called frond and root is called rhizome. So we got it. We got it from NBRI campus. We also explored Pachmali Biosphere Reserve. We prepared total soluble protein and we performed bioassay. I will talk about bioassay a little later. And then we selected a plant which caused extract which caused more than 50% of mortality of whitefly. Then there was a question we were hunting a protein which can cause toxicity. So the toxicity here could be because of any other thing. So we actually treated the total soluble protein with proteinase K and we gave the thermal treatment and then performed bioassay and selected those fractions which actually where the insecticidal activity was lost due to protease activity and thermal treatment. And then we purified the protein, which is toxic to whitefly. So, I'm just trying to explain this strategy. This is a plant. And then we prepare total soluble protein. And we then fractionated the protein with, ammonium sulfate. So ammonium sulfate salts out the protein differentially. We got several fractions. Now we had to test this protein against whitefly. Whitefly is a piercing and sucking kind of insect. We prepared a diet. We mixed the protein in diet. It was sandwiched in the 2 layers of parafilm and then allowed, insect to feed on this. And we found toxicity, and wherever there was toxicity, we took that fraction and tried to purify the protein to nearly homogeneity. And it happened. So we screened around 254 plants, 11 plants were positive, showed insecticidal activity. Then from 5 plants, we have purified 5 proteins, which actually we are deploying for insect resistance in cotton, and they are at the various stages of development. But Tma12 protein was purified from a fern, namely Tectoria macrodonta. This is a very, very unknown kind of fern, but it is an edible fern. It is found in Madhya Pradesh, in India and Nepal. It is consumed as a vegetable and salad, and its concoction is traditionally used for the treatment of gastric ailments. So we thought that we are going to isolate a protein which is, the source plant has, we have a history of using source plant as a food. So we are very confident that probably whatever the protein we get, that will get regulatory clearance faster. So let us talk about how we could purify the protein. When I'm talking, I am remembering Dr. Vinod Bhakuni, late Dr. Vinod Bhakuni, who taught me the tips of protein purification. So we prepared total soluble protein from rhizome and leaf, and we found activities in both. So we thought that let us actually target purification of the protein from leaves only. And then we performed ammonium sulfate precipitation, and we found that the maximum activity, insecticidal activity, was in 60 to 80% fraction. We took this fraction and we started optimizing ion exchange chromatography, and we could purify this protein as a single peak with almost 85% homogeneity. Reaching to this level was very, difficult. So we call this protein Tma12. T denotes bacteria, Ma macrodonta, and then 12 means we have attempted a total of 12 times to optimize this protocol. So it is called Tma12. So we purified to nearly homogeneity. So all the proteins here was very beautifully, fractionated, and we got this fraction. And then we purified the protein to homogeneity on size exclusion chromatography. Then we characterized this protein. This protein was toxic to whitefly at LC50 was 1.49 macrogram per ml. This protein has a single isoform and it exists as a dimer. But you cannot produce a recombinant protein in such a high level in plants like cotton. So at sublethal doses like 100 nanogram per ml, this protein interferes in the reproductive cycle of the insect. And it is exclusively toxic to whitefly. It is not toxic to any other insect. Now we got this protein. To this level of purity. Now we do not know what is this protein. So what actually we did? We did protein sequencing on MALDI-TOF. So what is the beauty of this system? So if you take peptides of a protein and you fragment from both the ends, you get a very messy data. The fragmentation happens from the N terminus and also from the C terminus. N terminus generates B ion and C terminus generates Y ions, and actually they make the data very complicated. So what actually we did, we blocked all the ions with sp-ITC chemistry. So only Y ions were allowed to enter in the time of TOF tube, and we get the clean data, and with this we could resolve some of the peptide sequences of the protein. And then we clone them. So this gene is 209 base pair long, and it encodes for an open reading frame of 651 amino acid residue. It encodes it is 651 base pair long. It encodes a protein of 216 amino acid residue. The precursor protein is transported to endoplasmic reticulum. A signal peptide of 24 amino acid is cleaved, and 192 amino acid mature protein is produced in the plant, which is toxic to the plant. But why this protein is produced in plant, we do not know. Then what is this protein? So we wanted to know that because the protein is quite unknown and it is toxic to whitefly, but it was important to know the structure of protein. So actually I collaborated with Dr. Prema Vasudev in CSIR-CMAP, and she crystallized this protein and established this as a lytic polysaccharide monooxygenase. So this is the structure of polysaccharide monooxygenase, and it has two intramolecular disulfide bond. It forms a histidine brace-like structure with the N-terminus, and it engages a copper atom to make the protein stable. The protein can be characterized as a histidine brace, and it has a glycosyl unit. This experiment was published in Planta in 2019. So when we published our work, transgenic work in Nature Biotechnology, there was a consortium of scientists in US and in Europe. They actually hypothesized that the gene cloned at NBRI has migrated from bacteria to the fern through horizontal gene transfer. Somehow, we do not agree with this hypothesis, and we are working on it. However, at present, the hypothesis is that the gene has migrated from bacteria or fungus to this fern, Tactidia macrodonta. We do not know the function of this gene and protein in the fern. What is very interesting that despite of very low homology, like 30% the three-dimensional structure of TMA12 and bacterial lactic polysaccharide monooxygenase is quite superimposable. I would like to inform you that this is a very, very important enzyme with the industry perspective, because it is used in digestion of complex carbohydrates to make alcohol. Now actually we move. So we got the gene, we got a promoter from Arabidopsis, we develop a construct, and we introduce the gene expression cassette in cotton. So when you introduce the gene expression cassette in cotton, it means the gene should have its own promoter, coding region, and terminator, which is very suitable for cotton. So we did this. So we developed transgenic cotton. That is the cotton is called genetically modified cotton or transgenic cotton. So we had used several promoters to express this protein. We developed several lines, and finally we selected 4 lines which actually showed quite high level of tolerance to insect pests. So this photograph shows that the non-GM cotton, which is fully covered with whitefly, and GM cotton was quite tolerant, and whitefly could not multiply on it. So you can see, eggs and nymphs on non-GM cotton, and it was very, very, small in number on GM cotton. So the protection of cotton from whitefly actually enabled cotton to produce good amount of cotton balls. So you can see it here. So we analyzed we got several plants, and then we studied transcript level, and we found a few lines with a very high level of transcript. These lines were tested for production of TMA12 in cotton. So it was analyzed by immunoblot analysis, and then we performed a trial of GM cotton. We challenged with whitefly, and we found that the plant that expressed protein at a very high level had lowest count of whitefly, and the plant which did not produce TMA12 in good amount, the whitefly count was higher, and control cotton had a lot of whitefly, as you have seen in the photograph. We took this plant to polyhouse, and so whitefly is a vector of viral diseases. So the idea is, if you want to control malaria, you control a mosquito. So if you want to control viral disease in plants, you control whitefly, viral disease will be automatically controlled. So here we have grown, transgenic cotton and control cotton, challenged with virulent whiteflies. We found that transgenic cotton is absolutely normal, and then you see, the curling of leaves in cotton, and these are the symptoms of viral diseases in cotton. So when virus infection happens, cotton plant becomes photosynthetically very active, and then it enters in the next cycle of plant and does not produce cotton balls. which is required by the farmer. So we tested GM cotton, presence of viral particles in control and transgenic cotton. We found that viral DNA was present in control cotton with its alpha and beta satellite DNA, which was absent in GM cotton. So this shows that another trial where either plants, in case of non-GM cotton, most of the plants died because of whitefly predation and fungal growth. The plants which survived got infected with viral disease and enter in a new cycle and started photosynthesis. On the other hand, GM cotton actually produced good amount of balls, which actually you can see in the photograph. So this is another trial between the a low expressing GM cotton and high expressing TMA12 expressing GM cotton. Very good yield we recorded in comparison to low expressing lines. And finally, these are the two lines which we shared with Punjab Agriculture University, Ludhiana, and CICR Nagpur for independent, unbiased evaluation. So the evaluation is under progress, and in next year, this is the last year trial. Next year, they will take a call to deploy this cotton for a variety development. So it is very important that when you introduce a gene in cotton or any transgenic plant, you need to inform RCGM where the gene has gone. So for example, in event 402, this gene has gone in a chromosome. Cotton is a tetraploid genome, and it has got in chromosome D08. So when gene enters in a genome, a part of DNA of the genome is deleted. It is possible that new sequence is also inserted there, and it is possible that the part of your DNA which you want to insert in cotton in a crop, that is also deleted. So we did all these analyses, and we found that our these events do not. We also need to know that when you introduce a gene in crop, it should not disturb any functional gene of the host plant. So we did not find any disruption of a functional gene of cotton. And so when you introduce a gene in a crop, you also try to see that the insertion did not create an ORF which can produce a protein which is allergenic. So we did not find any such protein. So we found that this plant, these events are quite safe, and we can use it for variety development.
I'm saying it's time to conclude.
I'm just concluding it.
Yeah.
So then I interacted again with CDRI, and we performed a safety study with CDRI, and we actually we learned that GM cotton, the cotton seed cake meal is fed to the calves. So around 450 grams of seed cake meal is given to calf. So the equivalent protein was fed to rat, and we found that the protein has no bad effect on the treated rat. So now actually, so we have introduced the gene in Cocker 312 variety of cotton, but it is not a cultivated variety, it is an American cotton. Now we have introduced the gene in a popular variety of Punjab, Haryana and Rajasthan, which is F2228. So the back crossing is under progress, and maybe in 1 year or so, the variety will be developed. So with this, actually, I am concluding that a whitefly toxic protein was isolated, and it happened to be a lytic polysaccharide monooxygenase. It is very insect specific. It does not cause any problem. When when we fed the protein to rats, and we hope that at some point of time this technology will be used by the farmers of country. So how it will be used? So it will be used like, you know that the BT cotton, and then we have a cotton, we call it as a Cry-1 EC cotton, and then this TMA12 cotton I talked about. If we stack all three events in cotton, the 3-gene cotton will be tolerant to 5 different insects, and we are running this project under Cotton Mission. This is Plant Molecular Biology, Plant Physiology team of NBRI, and I thank you very much for patient hearing. These are my collaborators. CDRI, I thank Smriti, Vahaj Bhai, Sarika, and Sharad Bhai for conducting testing the protein for its safety. And thank you very much.
Thank you very much, Dr. Singh. I think we are running out of the time, but still, if anybody has a burning question, one question.
So we could not take it properly. So when this plant will go in deregulatory procedure, all such things will be tested. So I do not know the subject. Actually, it is all the team CDRI who actually designed the experiment, and then they performed some experiments with. I understand question, and I had faced this question earlier also, but we could not do it. But it will be taken up in a proper manner.
Okay, Dr. Singh, thank you so much.
No, the calf is an example. So how much protein we should test against the rat? For that, actually, we took that as an example to calculate the dose. But in future, when the detailed deregulatory procedure will start, all these things will happen. Then GM seeds will be used for treating the feeding calves. No, they were not fed with the seeds. The calves are not. It is a rat for how much protein to be fed to the rats. We use that data, for calculation of the dose.
Okay.
People talk about developing resistance against, BT cotton, right? It has remained as a myth. BT cotton, stood for 20 years. So insect had to engineer itself a lot to develop resistance against a protein, right? It can develop resistance against pesticide because that biology is different. But if you ask insect to develop resistance against a protein, not so easy.
Okay, I think there may be questions, but they can be raised during the lunch time. So, Dr. Singh, I have one. First of all, thank you so much for really painstaking and passion you had to obtain the protein, purify it, clone it, and then finally inserted it and made it. I have one curiosity that we had a BT cotton. Now we will have this cotton against the fly. Now, is it possible to have one cotton variety having different genes? Theoretically.
Sir, it is my dream that I should be able to delete around 100 kilo base pair DNA from the cotton genome and stack 5 genes there to create a locus for insect resistance using the CRISPR technology. So we are working on it. It is too early to make any comment.
Yeah.
But it is an aim. It is possible that I may retire, but somebody in my laboratory should do it.
Good, it's possible. Thank you so much. And I thank organizers for giving an opportunity to share this session.
Thank you so much, Dr. Seth, for chairing this session, and to both the speakers for very interesting talks. We will now take a short break for tea outside. I think the light has come back, so when we come back to this audio, it should be cooler. So just a short break, 10-minute break for tea outside. Please join us, everybody. So welcome back to the second session. This session is to be chaired by Dr. Madhu Dikshit. Ma'am, please come up on the deck.
To the auditorium after a very, very interesting session in the morning. And I feel it is late for good morning and a bit early for afternoon. So good afternoon, everyone. In this session, there are three speakers, as someone has already pointed out. So without wasting any time, I will invite Professor Harshvardhan Vanare. He is from Department of Physics and the Center for Lasers and Photonics at Indian Institute of Technology, Kanpur. His interests range from classical to quantum aspects of light-matter interaction. After receiving his PhD from the University of Hyderabad, where he pursued the study of varied aspects of coherent light-matter interaction, he joined the Washington State University, USA, for a postdoctoral fellowship on semiconductor optics. He was also a postdoctoral fellow at Illinois State University, where he worked on light propagation in random media. He joined IIT Kanpur in the year 2000. He has published in the areas of coherent control of light-matter interaction, quantum optics, nonlinear optics, metamaterials, anisotropic media, photonic bandgap materials, light propagation in random media, and biomedical optics. His current research interests range from quantum and classical aspects of orbital angular momentum states of light, control of pulse shaping and propagation in structure media, imaging to create effective optics-based solutions for biomedical diagnostics. So I request to initiate your talk.
Yeah.
Thanks.
Thank you very much. Yeah. So first, I'd like to thank the organizers for giving me this amazing opportunity. And I feel completely out of place simply because you guys are looking at single amino acids connecting large DNAs and doing amazing things. I don't do any of this sort. We are looking at everything as a small unit and hope it works. So physicists in the wrong place, but bear with me for the next half an hour. And this is the title of my talk. Yeah, so life and sustainable life. I tried to connect the two. And my original idea was light and life, but then Dr. Habib said sustainable life would sound better. And hence I have taken that route. I hope you doesn't change which it does. Okay, there's a time lag. Fantastic. So these are the sustainable goals, right? And probably I will touch upon not really anything deeply, but about 8 to 9 of these. And I will stay true, hopefully, to the basic premise, which is basic sciences and sustainable development. We will see how we go about doing that. Right. So this is supposedly a light talk. So I'll start with photosynthesis. I will try to connect all of these topics which seem unrelated at the end of it. So if you give me enough time, I will conclude my talk. But before that, please do not. Anyway, that's a request in the chair. I look at light, its aspects, a few of them which are relevant to my this thing. And this is the main idea that I want to focus on regarding sustainability. And one can literally use the universe and the earth as a thermodynamic engine and hopefully do something useful passively, right? And the way forward is essentially hoping to mimic nature, and probably that's the way forward. So this is what is the whole sequence, and I hope I can convey some meaningful things. All right, so light is the source of literally all life on Earth, right? And I will probably dwell on one particular aspect that nature does variety of things in an extremely efficient manner. And we need to pick up those aspects of efficiency and put down in simple systems so that we can probably perform better. And as very rightly pointed out in the morning, I mean, the way we are destroying the Earth in terms of its resources, I think we need to learn from nature how to sort of sustain itself and in a sustainable manner. All right. So, all right. Okay. So there is a delay, and I need to probably point it in a particular direction. Oh boy. Yes. Sorry. All right. So, for example, the green color, right? So you know the green color arises because of selective absorption. So largely there are variety of photosynthetic, photoactive molecules in a leaf, right? And it's chlorophyll that basically gives this green color. So what it does is basically absorbs light in the bluish region. This is about 400 nanometers or 0.4 microns if you want to think about it, and also in the red, right? It absorbs these parts so that what is remaining is the green part, and that's how we look at, we see green color on the leaves, right? So the question one is asking is the following. If you look at nature, and it appears as though there is a lot of greenery around, right? So is nature overindulgent when it comes to greens, right? It appears as though nature is overdoing it, and I want to convey to you that it is not really. So let us get to some interesting, the mechanism. So basically, there are, I don't want to tell to the audience, which is specialists in biology, there are these chloroplasts where there are chlorophyll molecules stacked in these discs and so on. So this is where the conversion of light happens, and the typical concentrations of chlorophyll in leaf is about 400 micromoles per meter square. Correct me if this number is wrong, but this is an average number. So if you think about it, you convert these moles into molecules, it's about 10 to the power 20 molecules per meter square, which is there in a leaf, right? So what do we compare this with? We compare this with the typical solar flux. So solar flux can vary from 1400 watts per meter square, very bright light, to average 700 watts per meter square. And then you calculate the number of photons per unit area, which you can do a very simple calculation. Go by this formula. You will see is about 10 to the power 21 photons per meter square. You see the matching between chlorophyll molecules and 10 to the power 21 photons. That's absolutely amazing. Now let me tell you why this is important. I am a quantum optics person. I look at something called the quantum aspects of light. What it means is that, for example, the motivation for learning quantum optics is that the light we see is not really continuous. So if you put a detector and you lower down the light, it will go tick, 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 and so on and so forth. It is actually quanta by quanta, and we perceive it as a continuous light. So question one asks is, when is it, as I said, tone down the light, decrease the amount of intensity, then you see quanta. That's not the point. At the very basic life level, every photon matters. This is one-to-one correspondence between photon, availability of photon, availability of the chlorophyll, and all that stuff, right? Photosynthesis efficiency is known to be 30% for making the glucose. I'm sure the tree does a lot more. I just came to know from the previous talk that tree does a lot more to protect itself. But then if you look at that efficiency is 30%, but there are about 23 steps which are happening, which need to happen at 95% efficiency to get to that number of 30%. So please understand that there is nothing wasteful about nature. It is just right. This is one aspect. So let me now change gears and just tell you that, this stuff is really true. I heard of something called the Young's double slit experiment. You put light through, you learn it in the school. I probably hated it. But what we can do the same is that to put through the Young's double slit experiment, this very low light levels where you think quanta is important, in the interferometer, this is the interferometer through which light goes through. It has a path to choose left or right. But in this interferometer, it actually one photon at a time. It doesn't know the other one is arriving. And you see that the photons form this individual photons. If you collect and keep their history, you will see this interference pattern. Right? So that is essentially the nature of the very quantum aspect of light. And that is very important. It's important even for sustaining life on Earth. Right? Yeah. And then, we worry about other things. We worry about statistics. For example, how are the photons arriving? If you look at a light like this, the photons come in bunches. If you look at atoms, single atoms emitting light, they never come together. Then if you look at a light from a laser, which is like this, then at the top one, they come randomly and so on and so forth. Some details which I don't want to get into, not relevant at the moment. Now I switch my gears and go to eye, right? And I'm sure you know more biology than me, so I will not belabor the point. This is a familiar curve, familiar sort of schematic. I'm interested in the retina, by the way, also the lens, of course. So I'm looking at it as a physics problem, right? So what you have is an amazing detector. So by the way, we are not able to mimic the eye when it comes to detectors in real science. I mean real science in the sense that the science that we do in laboratory. So for example, our eyes work fantastic outside, inside. The dark adjusted eye, by the way, can detect roughly a photon flux of 5 photons per second. That's survival. Understand? So even as little as a 5 photon per second will be a dream detector for a single photon detector manufacturer. And our eyes do it simply every night when you shut off the lights, right? When you see this, you have the feeling you're walking that you have somebody following you. It's probably nothing sixth sense. It is just eye measuring a very highly, a single photon probably falling from a very large angle, and you think that you have, probably somebody behind. So that's survival. And light and eye does it beautifully. I want to get to some other number. So apart from this, which that we make an inverted image optics wise, but our head that allows it to turn it around. And by the way, people have done experiments where you continue to have an inverted image, and then within 20 minutes, our brain adapts to behaving the way it is. Right? So brain is an amazing beast there. So let's not get there. So anyway, there are 3 particular types of, so there are rods and cones. I'm sure you know the story. There are cones of 3 different types, blue, green, and red. Relative firing of these tells you these other subtle colors that we make. So the color I see, the color you see, you remember that big debate in on the internet, that blue dress or the golden dress, it has got to do with your eye. Okay, so it's your dress. So anyway, so there are these light sensitive photoreceptors. Literally, if you come to, let me get to the detail. So there are these cones and rods, as I said. The cones are largely centered at this region called the fovea, right, just behind the lens. So cones are responsible for color. One doesn't want to do color when you're surviving in a forest late, dark in the night. You want to appreciate color when you're lighted up. So it's roughly there. And then rods are everywhere. Rods are the sensitive ones which are sort of color immune. They do have a response, but then they are more sensitive than the cones, and they are spread all over the eye, right? And there are these, this itself is a fantastic step from light falling here all the way to detected there. How is it happening, right? So that is another story. At the end of it, you require a lot of blood in the back of the retina so that light photons don't bounce around in the retina so that you get blurred images. It needs to absorb it. So it's just an amazing, absolutely perfect device for detection. So the difference between owl eye and our eye, owl sees better, is only that they have a much larger retina, double the size. Okay, and because they have a larger retina and they want to actually figure out which way the photon is coming, they turn their heads beautifully. Right, so zero in. Okay, so now that I come to eye, I want to just get aside to imaging. What is the imaging? Imaging is a place where if you want to get a Nobel Prize, work in this area. Nobel Prizes are peppered all over the place. So I will not belabor this. You guys use the microscopy all the time. What is the main issue? Issue is the following. I have an object, and physicists are extremely lazy people. They actually draw arrow to show a lens, by the way. So this is a lens, I believe. So we have an object and we form an image. What is the image? It's a point-to-point mapping from the object to the image. So what I want to do is I want to make a image of that point back there, right? So it so happens because of my finiteness of my system, I will basically map a point, I've exaggerated here, into a disk. Okay, this is called a point spread function. And this size of the disk depends on the wavelength of light. Okay, and also the aperture size of your lens. So bigger the lens, finer the point would be, right? Look at the angular size of the disk, and it depends on the wavelength. The larger the wavelength, the worse it is. Blue light you will see slightly better than what you would see in red light. So this is the point spread function, and the resolution. That is, if I take 2 points, because if the points are mapped to disks, the disks are overlapping. So now we are asking, can I resolve these 2 points? Would be that, can I resolve these 2 disks as independent? That's the job of the eye, right? And nature does it beautifully. So I will not get into this. You can do better than the resolution. I thought this physics talk, I will not get into details. But if somebody has a question, I will tell you how this works. But apart from that, what you want to do is you want to, basically that disk is lambda square, by d. You want to make it better than lambda square. Can you pinpoint? Yeah, you can do better. That's on the point. Let's get back to the eye. So look at the sizes of these rods and cones. This is a SEM image. I don't know why one would do that, but we take an image and we find out the size of it. And this is 2 micron square. Okay, that's the disk. I'm talking on the top there. I'm not talking the length of it. The detector is the surface, for example. And look at, so I calculate the delta theta, which is the point spread function's, angular size. That's about 1.64 10 power minus 4 radians. This is in units of, degrees radians. This is radians. So if you look at the size of the disk, is basically 20 mm is the size of a typical eye. So this is 3.4 micrometer. 2 micrometer, 3.4 micrometer square. You see the matching? Amazing matching. And by the way, this is for 4 mm. Our eye, the iris, which controls the amount of aperture size, like in a camera, it goes from 2 mm to all the way to 8 mm. So there is some adaptation. So this is just a qualitative number. But look at this matching. There's no point putting more pixels. It's wasteful. Nature does it beautifully. You can't have very large eyes, so it's adapted to that size. All right, so now I'll come to a few other types of light, which is color. So I'm interested in all these frequencies, wavelengths. This is something I picked up from the internet. I'm sure you can have a look at it. We are looking at visible here, right? That's what I talked about till now. And I will look at something called in the mid-infrared. All right, I'll talk about it slightly later. But then that's the region where, so variety of sizes. The way we think about light is, okay. When we were in school, light was in rays. When we were in 12th standard, it became a wave. When we came back to BSc, then it became sort of wave with much more complication. And then we went to masters. They said, no, actually it's a particle, a photon. And then we get there and we say, actually nobody understands. Don't worry about it. It's both particle and wave, and we are happy. Nature doesn't like to distinguish. We are stuck in our heads, so we like to distinguish and so on. So that's one aspect of light, which is the color, the frequency, the energy carried by the photon. It's very important. So, for example, if you had the planets away from sun, the colors of leaves might have been different. Just the way we look. It would have been darker plants if you're a little further away, because then you'll have to color that you're, and so on. So polarization is another aspect of light, which is what makes it vectorial aspect of light. So you can have a linearly polarized light where the electric field is going like a circle, that's circularly polarized light and combinations of thereof. And you can see all this in these glasses that one makes, Ray-Ban glasses. Have you seen that? So for example, you have a trough of water where you have got light falling in from the sun, and then you have stones underneath, right? And you see that at reflection, the light basically, one polarization reflects differently from other polarization. So you see that one can basically put a Ray-Ban glass like polarizer cutoff thing, and you can see light coming from inside. Now, this is not a trivial example, by the way. Raman figured out that the ocean is blue because of this experiment doing on the ship. See, that is what makes us different from him. He was traveling in a ship. He had nothing to do, so he was carrying a lot of optics in his pocket. He was an expert, world expert in acoustics, but he was carrying stuff for the optics. He figured out why the sky is blue and then went on to do a lot of optics. That was the beginning, and this is the experiment he performed. That's another story. All right, so this is my most important side. I'm coming to now Earth as a thermodynamic engine. So these are, 2012, this was published in the IPCC report, and this is what tells you how Earth functions. In the end, we are living in comfortable temperatures, not too, but otherwise 300 Kelvin, more or less, Earth remains more or less stationary. How is it possible, right? So what you have is, just look at these numbers. Sorry to be detailed, but then the point is in the numbers. So 341 watts per meter square. So these numbers are all watts per meter square. Coming in from about 79 watts gets, what meter square gets reflected from clouds, another 23 from the surface. So you see that about 1/3 of it is just gone away by reflection. Okay. 161 is absorbed. About 78, the equal amount that is reflected is with the atmosphere. Right? So your atmosphere that is taking away this energy. What does the atmosphere do? So every object at a temperature T finite radiates energy. This is called the Stefan-Boltzmann law at some sigma T to the power of 4. So everything that is at a finite temperature radiates. It has got a characteristic radiation pattern. By the way, we thought we understood physics and systems very well. Pre-1900, Planck figured out this law because this was not explained otherwise. Hotter things were looking more bluish, whitish, and so on. So that is a different story, but every object at temperature T emits radiation, right? Fine. So sun is at 5,000 and odd Kelvin, so it emits. Now we have got energy which is stuck in the atmosphere, right? And it falls on the earth. By the way, there is evaporation happening, there are thermals happening, but this is the energy that is circulated back and forth. So there's nothing to talk about as an overall engine. And then you see that you have the surface radiation, which is going out, some of it getting absorbed again, and these greenhouses are reflecting back. So be careful, because the atmosphere is getting not only from the sun, it's also getting from the earth, so it's emitting everywhere, both sides, top and bottom. At the end, you add up these numbers, 239 goes out there, 102 goes out there, and add up these is 341. Actually, it is, if you put the numbers right, it's about 0.9 watts per meter square is remaining with Earth. Okay, 0.9 watts per meter square. That's the amount which is remaining, leading to sort of heating or whatever. Right. Now that we know this, I am interested in this. There is this 22 watt per meter square which seem to go from the surface of Earth all the way out without as though looking at the atmosphere. Okay, and that's something which I want to focus on as I go along. So this is the solar spectrum. I'll try to be as pictorial as possible. So this, I said, this is the typical theoretical curve as described by Planck. He was so good, and that matches with the sun's spectrum at 5,250 degrees Celsius. And by the time the yellow thing is on the top of the atmosphere, by the time you come down, then a lot of these gases have taken away some light. So what you get is this red curve with a lot of dips and all that. Each of these dips correspond to absorption by that particular species. Now it so happens that nitrogen and oxygen are abundant, but they don't participate in this part too much. Okay, so now you're looking at, of course, the numbers you see is largely the radiation is confined from 250 nanometers to 2,500, or in my words, it is 0.25 microns to 2.5 microns. That's the range in which solar influx is coming. Right, so people talk about energy, light-based energy. I will not touch into any of this. Not interested. I mean, I am not interested at the moment for the talk, but very, very important. Clean energy does hell a lot. We should have done this earlier. But the manner in which we are doing also is not, the other issues with probably Venkatesh, I will leave it to him to discuss how we are doing it badly. So anyway, so I'm looking at something called radiative sky cooling. I hope I have 15. Thank you. Okay. So this is the main slide. So I have this input radiation that is coming in. This red one is the input radiation. Of course, that is coming in, and this much is falling on Earth. But what Earth does is, it takes away this radiant energy, converts into heat, and then it's emitting it back. So it so happens that there are of course various gases. For example, water vapor, you see, it is absorbing all over the place. And then carbon dioxide. This is in wavelength scale. So from 0.2 microns, you see that 0.2, or let's say even somewhere, this here is about roughly about 0.35 or 0.4 microns, all the way to 2.5 microns. That is where this is, right? This is where the input is coming from. And this part is the one which is the outgoing thermal part. Now, why do I say it's outgoing? Because you see that if you look at like I said, that if you have a typical temperature T, an object, it will emit radiation characteristic to that temperature. It has nothing to do with the material. It's got to do with the temperature. So we are roughly at 300 Kelvin, and that black body bell-shaped curve at peaks in this infrared region. It's called a mid-infrared, and it basically peaks between 8 to 13 micrometers. Okay, very, very important. And it so happens that there are very few that actually contribute to absorption there. Okay, so this is the breathing mode of the Earth, which is trying to give out that energy and keep itself reasonable. Is that okay? That's very, very important. So you see, now, by the way, all these gases, they are very, very important. For example, the ozone takes care of the UV. So, we don't get that. Then Rayleigh scattering largely happens here and so on. So if you look at the total absorption and scattering from the atmosphere, you see this little window where there is little happening there. Of course, it is compromised. So you'll see why people are harping about carbon dioxide, because carbon dioxide has a little remnant there in that 8 to 13 micrometer window, right? That's a window for Earth to cool directly. And that window, if you play around with, you're going to temperature of Earth. So you see, carbon dioxide limits it. There is a little bit of ozone, which is, by the way, very little. It's up there. And then a lot of water. So if you have a very cloudy situation, very humid situation, in that case, you won't cool as efficiently. All right. So now this is the distribution of the greenhouse gases. As I said, nitrogen and oxygen largely transparent in the discussion that we are having. All right. So now that we know this business. What is the business? We are incoming here. This is a visible window, right? 0.3 now, relevant window, because we are not looking at top of the atmosphere, looking at on Earth. 0.3 microns to about 1 or 2, let's say, that's the incoming solar flux. And this is the atmospheric window where nobody in the atmosphere participates, apart from ozone, which is little far away and too little to bother us too much, right? So typical thermal systems are these. We have been doing this all the time. Take the solar energy in, convert that into heat, use that heat for some output. There is some remnant here, there is convective thing, this is the radiation coming in, and so on. There is some infrared emission and so on. This is what we usually do. We want to convert these systems, which are essentially in terms of units, same set, but need to operate very differently. What I want is, I want radiative cooling. That is, I want a radiator that will not take this energy, right? It will emit a lot of it in this range because nobody else is picking up in the atmosphere. And the rest of the system, and that cool part will be used for comfort, right? And so on. So this is precisely what I intend to do, and let's see how to go about doing. Now, this little bit of mathematics just to make things right. I cannot see if I don't do too well without the signs being correct. So this is the radiative part, which is what I want to radiate out. Atmosphere is putting in energy into my surface. Solar is directly coming in, atmosphere is putting in, there is a non-radiative, there is convection and conduction that is happening. And I have this body at, let's say, ambient temperatures 300 Kelvin, and out there is the universe, which is at 3 Kelvin temperature. So by the way, the cosmic microwave background radiation is at 3 Kelvin. We know that, we have measured that. So you see, you are looking at, if you look at 300 Kelvin power of 4, right? 10 to the power of 8, and this is some trivial number. So it's a very large difference when you are looking at radiative cooling. Then another equation, just bear with me. So if any object, right, it can transmit light, it can reflect light, it can absorb light. Okay, So this transmittance, reflectance, and absorptance, they all add up to 1. Nothing else can do. So, and it so happens that the amount you absorb is the same amount you emit. This is by Kirchhoff. Very, very important. So it is counterintuitive that I need to absorb as much to emit as well. Okay, understand this? Not easy, but that's why Kirchhoff's name is there. And it so happens that the amount of light which is radiated, it depends, as I said. So I have an object which is a temperature T object. It is radiating. There is a other ambient which is radiating back. So you see that the effective radiation is just this, right? That is what we are talking about is this ambient for a problem being in comparison to the 300 Kelvin that we have. All right. So this is the area that I work on, metamaterials, but then I have never really worked on this pure thermal emitters per se, I should confess. But then after preparing for this talk, I am leaning towards that side. Yeah. So what you want is solar influx, you want to reflect it off. That's from 0.3 to 3 microns. You don't want anything to do with it. So it doesn't even absorb. And then thermal radiator, I want from 8 to 13. That means I want to make this material, which is extremely emissive from 8 to 13 or absorptive 8 to 13 and extremely reflective between 0.3 to 0. That's my input, that's my output. Rest of the spectrum, I don't care, right? So, and have people done this? So metamaterials is what we talk about. So let me just tell you a little bit about it. So a material's response to light, I'll take 5 minutes. It's okay. So materials response depends on permittivity and permeability. That is the response to electric and magnetic fields. And earlier these were God-given, but then some people thought that you can do better, and we can actually change them around, even change the sign of the refractive index and so on and so forth. These are called meta materials because they are not which are available naturally in nature. So the deal is this. Again, apologies for being pedantic. So there is this Maxwell's equation. These epsilon and mu are actually coefficients that determine how we interact. We can change the signs of these in a very funny way. For example, energy is propagating. So wave is going, the wave phase falls and the energy flows in the same direction typically. But we can turn around things and make things quite funny. But this is the regime of metamaterial. Let me just tell you how you do it. So for example, you look at permittivity. For example, in a metal will largely reflect light. But you can also make the metal transparent. So what it does is the reflection, the amount of light, if you know, this is a complex number, but in omega is sort of, above a certain frequency, which is omega p, this becomes like a dielectric. Below that, it becomes like a heavy reflector. So what you can do is you can change the number density of electrons by taking a material and taking thin wires. So these wires are sizes which are much smaller than the wavelength of light. So the wave sees only an average material. That's what is the notion of metamaterial. Similarly, for example, there are no magnetic resonances beyond gigahertz in usually God-given materials, but we can make magnetic resonances at gigahertz, for example, where we take a material, make these little rings, which acts like a L and a C capacitive gap and C resonance, and this can be made to sort of hertz and so on. So one can play around and change these properties so that one can have epsilon and mu as one likes. So this is something that in microwave, in optical, depending on the scale, one does various things. These are just two cold bars put in a particular direction for a particular polarization of light. This will give you the characteristics that. So for example, this is something that we did, which is to make a metal transparent and so on and so forth. Let me not get into that. Let me come to the main point. So radiative sky cooling again. So what people have done is the following. People have taken these stacks of materials, okay, such a way that they have got the characteristics that you want. You want emission at 213 and you want complete reflection at, and you see that many of them are triangles, and I'll tell you why. Many of them are triangles which are, and these have been shown. What is most interesting is daytime sky cooling. I'm talking about daytime sky cooling. People have got temperatures below ambient to about 5, 10, 25 degrees Celsius. That's absolutely amazing. All that you have done is you have kept structure which is reasonably well engineered. That's it. And it just cools down. We are talking about daytime sunlight. And this will, for example, this is just a polymer film on which there are these transparent glass balls, silicon dioxide balls for particular size. Okay, some amazing work going on in this direction. So the way forward is biomimicry. I'll just take last one slide. There are these Saharan silver ants, they are very smart. So they survive in direct sunlight, do it very well. And bingo, what they use is the hair on their skin. And they have got a particular profile which is triangular. And it so happens that if you look at their, all over the visible, they kind of reflect. These guys have actually removed hair from the sample and with hair and done experiments, something cruel. I don't want to worry about that. But the hair that sort of makes it completely reflective. And in the mid-infrared, look at this transition point with the hair. You see that between 8 to 13, it actually uses the atmospheric window to cool itself, right? Probably nature knows best. And hopefully I have put in these all aspects together. We need the efficiency of photosynthesis, the scale accuracy of the eye, integrate all the aspects of light from color, polarization, maybe quantum aspects. And then, use this Earth universe system as a passive cooling mechanism. And by the way, the amount of energy used for cooling in the West is 60% in the US goes for maintaining buildings, 80% in Europe. So you understand that this is, and we require to cool, as we know today. And way forward is probably biomimicry. And it is not just one place. It's not just Saharan silk insect, silk ant. What did I say? Saharan silver ant. There are other species that do this with great elan. Thank you very much for your attention. I look forward to your questions. You are not, I think. You have to press, yeah.
So it is, no, but it is not working. It is working now? Yeah. So thank you so much, Professor Harshvardhan, for your very interesting talk. You have touched upon various topics within a short span of time.
Yeah.
And if there is any question from the audience. Yeah, Dr. Singh, yes.
Absolutely. People are looking at paints, by the way. Apply that paint and you will cool immensely. Yes. That is correct. That is correct. Why automatically? So automatically it so happens that on Earth, we are terrestrial. So terrestrial temperatures, there is, so even if you cool, for example, I cool other wavelengths, the atmosphere will give that back to whatever I on me. So the 300 Kelvin is there, but there is a window of opportunity, which is that atmospheric window where if you emit, atmosphere is not participating in that top. It is not stopping it. It's not absorbing. There is no more. Of course, if you have a extreme and sort of compromised. Yeah. No. Really. See. I mean, we evolved. Participate in these. Nitrous oxide does. Oh no, so okay, so we are people. What we do is even if a simple molecule which has got various energy levels. So moment you confine things. So for example, in an atom you confine electron to the nucleus. The levels allowed where the electron can be is not continuous. In a particular stack of levels, quantized, they have only certain energies. So when you take nitrogen together, they cannot have the individual levels of nitrogen or oxygen. The collective levels will be different from either of these, and they will form a set of bands. These bands, O2 in this, but they do. I mean, that's the way nature is. We can calculate.
So I will like to interrupt here.
Yeah.
Maybe this discussion can still continue
yeah.
during the lunchtime. So I request now Professor K.S. Venkatesh, who is from the Department of Electrical Engineering at IIT Kanpur. He did his MTech and PhD from IIT Kanpur. His research interests are signal processing, image and video processing, computer vision, and its application in robotics, signal and system theory. He works on miscellaneous theoretical problems, locality invariance theorem, several theory, hyperprocessing, and law of non-diminishing symmetry. He has 8 patents, more than 130 publications in these areas, and he shall be speaking Are Science and Technology Inherently Unsustainable? So may I request Professor Venkatesh to deliver the talk.
So this talk is going to be very different from the very technical presentations you have had since the morning. And what I wanted to address, this actually is not the field of research of mine. You're hearing the first non-expert talk since the morning. So, okay, since this was about sustainability of science and technology, I realized I have just been thinking about this. I dabble in this kind of stuff. So sustainability itself has many facets to it. One, of course, is, I think the most important one is the sustainability of intellectual progress. When we are talking about whether science and technology are sustainable, we need to look a little more closely to see what we are not mixing up with what. So first of all, there are several things. One is, all this climate change and pollution and whatever, depletion of resources, that's on one side. But more importantly, there are other questions about like, can we keep on inventing? Can we keep on understanding, progressing our understanding of nature at the same rate that we have been able to exhibit in the last 100 years or so? You can already see that since 1940 or 1950, considering those 50 years of the 20th century, physicists really haven't showed much progress in the last 75 years. When I asked one of my friends this, he said, oh, that was just a one-off. It doesn't happen all the time. That was very, very lucky that we did so many things in such a short time. Okay. The second, and this is very, very important, it's not just enough to be potentially able to make scientific progress. It's equally important that there is political popular support for it. If it doesn't happen, then we will be back to the Middle Ages and Dark Ages or whatever, where only people with a lot of money, landlords, etc., did some experiment in their homes for entertainment. And that's how actually modern science started. But it had to start like that because there was no governmental support, there was no popular support for it. Okay. Just one second. So that's very important to recognize. The third, of course, is the quality of life issue, all about pollution, etc., which we will come to. Now, what I want to address in these 3 parts, which will actually split up into a few more parts, is when we think, as we do today, that the most important dimension of human activity is scientific progress, it's also probably not very fashionable to talk about the weaknesses in science. And so I thought, let me spend a little time telling you about the shortcomings of the scientific method. These set of shortcomings are quite independent of who does the science. It could be us, it could be Martians, it could be anybody, but the scientific method has some inherent shortcomings. And they are the following. It's certainly no individual pastime as it is practiced today. It needs collective action. Requires a complete commitment and support from government and the public. It's practically a way of life as we have been practicing it all these couple of 100 years. When practiced with such thorough dedication, it becomes an end in itself. It has been estimated, and there's a very important point, that 75% and above of all scientific activity is only for self-sustenance. And that's what I mean by that is basically this. To sustain a nuclear energy program, for example, it requires you to train a huge number of sophisticated technicians, scientists. You actually start an industry to build instruments for the nuclear industry. Then you have technicians, you have academic institutions to train the technicians. You have to do all this just to get this little final product called nuclear power. So behind that little thing, behind that little output that you get, there's this huge investment in knowledge gathering, in manpower, in development. All this is essential. You can't just get the end result without doing all that hard part. And that's why I said that a lot of this is self-consumption, self-sustenance. Now let me go to the next. So here, what I want to say is, some want to point out the difference between engineering and technology on the one side and science on the other side. This is also going to impact our questions on sustainability. So scientists develop conjectures and theories and validate them in carefully controlled, closed or so-called laboratory conditions. Careful control is required to limit the number of variables that can affect the result. This is presumably necessary to assert or verify the independence or interdependence of various variables. So this is the only purified, sanitized kind of environment in which basic facts become clear. If you want to explain Newton's laws of motion, you would be told, look, it's happening all around you. You don't need a lab to understand it, but actually you do need a lab to understand it, because you have to make those frictionless surfaces, you have to use those billiard balls, you have to use those inclined planes. Why do you do all those things? Just because for us to comprehend what is actually manifest around us, it requires that we create situations where things are separated from one another. Otherwise, there are too many factors affecting every phenomenon you see around you. And so the pure theories don't come out of it. So on the engineering side, however, the game is very different. This is what I'm coming to. Engineers work in an open environment and not closed environments. In general, one has to be prepared for all foreseeable variables. One just cannot assume that some will not be there. The best we can do is to promise a certain high enough probability of successful performance. Certainty cannot be assured. Did it change? Okay. Fundamentally, it may be said that science plays the role, and this is the other relationship between science and engineering. One, as I said, is about science working in a closed environment and engineering working in an open environment. The other is that what engineering does is actually take off where science leaves off. Science tells you what you cannot do, what you can do. But just telling people what you can do doesn't completely, sorry, I messed up this thing. Just a minute. Yeah. So the laws of science do not limit you to one particular or one unique way of solving a practical problem. It only says you can do a lot of these things and you can choose which way you want to solve this problem, but it has to basically obey these constraints, conservation of energy, whatever, any of those constraints. However, the task of engineering is to select the most appropriate or the most convenient, the most profitable, more nowadays, way of solving that problem. And that's where there is a lot of more parameter choosing to be done by engineers, because we have to solve God knows all kinds of constraints. See, engineering needs to provide concrete implementations of systems that respect these constraints, of course, but the freedom of to choose from a variety of approaches that lie within the set bounds of the parameters. There may therefore be myriad different ways of solving a problem. We have to select the best one on the basis of a lot of essentially non-scientific considerations: cost, technological maturity, local material availability, environmental impact, expert opinion. All these things will come into finally building something in a certain way or doing something in a certain way. It might be water treatment, it might be power generation, whatever. Okay. Yeah. So now I come to some of the shortcomings, and it is this. The ability of the scientific method to make perfect predictions is limited for several reasons, and these are the reasons. The theory or model may actually apply only in a certain parameter range, though we wouldn't know about it until we observed a serious deviation. It's like in old mechanics, we had Newton's laws, which turned out to be not perfectly accurate with the with the theory of relativity and all that, but everybody was happy with it because we had not conducted those experiments, the kind of experiments, the level of experiments where we would have seen that something was wrong, that things were going off the scale. So we today have a model, we think this is okay, but this is okay only because only as far as you have validated it. You might, in a more thorough validation exercise, find out that it's actually not good. So this is the point that I wanted to make. So this is one thing. Until we observe a serious deviation, we may be carrying on mistakenly believing that it works on a wider parameter range. Simplifying assumptions is the next thing. You see, whenever you propose something, you always have to, whenever you build something, you always have to simplify a lot. If you don't simplify, things are so complicated, they just cannot be worked out. So we always choose to ignore maybe the effect of air currents when you do Galileo's experiment and say that, oh, it's only gravity. Forget about the air affecting it and so on and so forth. So these are the kinds of simplifying assumptions you make. So what happens when you make simplifying assumptions? You are deliberately choosing to be inaccurate. And that, of course, is a careful choice. You normally believe that the inaccuracies are within respectable bounds, but there might be situations where the accuracy goes off. Okay. Next, there may be known loopholes in the theory itself that make it useless under certain conditions. You must have heard of the three-body problem in physics, where there is a problem in predicting the result of an experiment. The theory itself is perfectly right, but you can never carry out an experiment which exactly does what you are expected to do. There will always be minor perturbations, minor deviations, which will completely change the consequences from what you might predict. So in short, the theory is all there, but you still can't predict what happens in a particular experiment. And our real life problems, mechanics problems, for example, are not just three-body, they are multi-body. So you will have so many situations where you may have all the equations right, but still, in certain situations, you cannot predict what's going to happen. You just cannot predict what's going to happen. Disproportionate or nonlinear consequences, systems exhibiting chaotic behavior, behave drastically different under infinitesimal changes of input. So this is kind of highly unstable systems. You do a little more of something and the result takes off in a completely different direction. These are all limitations in spite of having a theoretical model, having a theory about things. And these are intended to tell you that our inability to foresee all possible scenarios under open operating conditions will come up again for human, in the context of human lack of omniscience. See, we ignore certain variables knowingly. I have mentioned that already in the third bullet, simplifying assumptions. But then that's different from not even expecting certain things to go wrong. if you look at most of these nuclear incidents meaning unpleasant incidents, then you will see that you ask the people, why did this happen? They will say, we never thought this will happen, but it happened. So exactly, it's like asking somebody, can you think about something that you have never thought of? Or rather, can you think of something that you cannot think of? You can't. You cannot think of something that you cannot think of. And nobody would have expected there is an earthquake, then a tsunami, then a flooding of the generators, and therefore the reactors going out of control. That nobody would have expected. Now we are all wise, but it had to happen once. And in all future projects of science and engineering and technology, this will always be staring at you. You can never be completely confident that you are not going to do something seriously dangerous. It could always happen. Okay, now, so this brings you to the tentative nature of science. This is not to shoot down science, and I will make that clear in my concluding slide. This is to say that what we have is still something which is not as perfect as we think. As I have said here, all these above contributing factors or should be contributing factors to make mankind more tentative about its relationship with science and technology, and its reliance on science and technology to deliver heaven on earth. Oh, sorry, slide change. Yeah, sorry. Now, unfortunately, most in the scientific community either genuinely don't know or prefer to overlook or understate or blindly or simply blind themselves to all these fallibilities. It must be noted that these pitfalls are on account of the knowledge system itself, not the limitations of the practitioner. The knowledge system itself is this. You can be the most, the most effective way of propagating or practicing the knowledge system. These things will still happen. Now, even an omniscient being doing science would face every bit of this. Now we come to something which is, time runs faster when you're on the podium. Okay. So there are several limitations, and I'm not going to even read the whole slides. There are actuatory and sensory limitations. For example, we see only a small band of the electromagnetic spectrum. So a lot of what is here is not even being seen by us. It's just out of our range. So there is the second thing. The second bullet is about limitations of perception and computation. Oh, sorry. Yeah, I'm making this. Yeah. Okay. So there is this concept of bounded rationality that people in psychology and management talk about. I'll tell you what it is. If you tell something very complicated to someone, they will not try to convince themselves completely that you are right. They will do a rough calculation and say, yeah, I guess he's more or less right. Same way, I mean, this is about simplifying by dropping some variables, you can say. But the fact is, your rationality is not infinite. You are not infinitely rational as a species or even as an individual. You do the best you can because you have to take, it's like answering maybe an examination paper. You do the best you can given that you have to come out with the answer within 3 hours or whatever. And you say, this is all I do. So this is a limitation of us because we are not infinitely smart. We are not, we don't have infinite sensory perception, sensory capabilities. We are not infinitely smart either. So this brings us to the third thing, which is that very often in many practical situations already being faced today, as I have mentioned here, a pilot flying a complex machine like an aeroplane, there are so many things happening in front of him that it's very easy to do something wrong. In fact, they do it now and then and we have a crash. Something goes wrong, and just as you're trying to figure things out, because you need time to figure out, and they actually have little manuals there. They open it and see, okay, if this thing starts beeping, then I should be trying this first. Can you imagine somebody actually has a time to open a book when the plane is coming down or something? But the system is so complex that you can't carry everything in your head. And that is one of the limitations of the human being. As systems get more and more complicated. Did I change? Yeah. Okay. So the third thing that we have is a lack of reliable memory. This is something I didn't notice recently. Psychological studies have shown that memories in human mind are more like impressions of observations rather than reliable records of facts. Therefore, for example, memories of an event can be manipulated post facto when what one honestly believes he remembers can actually be a fabrication. Okay. Now, an often overlooked physical limitation of our species is also a limited lifespan. And for me, I just thought about it while preparing these slides. You see, we have a lifespan of whatever 80 years, and we are actually very productive and very receptive and capable of learning for maybe 30% of that time. And as science keeps progressing and there is more and more knowledge to be picked up, every child, unfortunately, you can't insert an SD card into his head and say, okay, you start from here. You have to teach them 1 + 1 = 2 to every child. You have to do it 1,000 years from now, unless by then they can figure out how to put an SD card in your head. You have to start from the beginning, but you have got a longer way to go because science has gone further. So what do? We have a kind of breakup of our education system into some common education where you teach them some 3 hours and all those things, and then you have specialized education. We end up with a lot of more and more narrow specialists in various areas. And each specialist cannot really communicate with the other very easily. It's not very easy for someone to explain pharmacology to me. And I guess I would take a very long time to understand it. I've tried that with mathematicians. No, I just don't get it. It takes a long time. It almost becomes, each subject practice becomes like a way of life. So as this goes on, as our lifespans more or less remain the same, do you realize that this is going to be a serious impediment to the sustainability of our technical progress? How much more can you keep cramming into somebody's head as it gets more and more? There is this joke about physical review. Physical review is supposed to be the most famous, most respectable journal in physics. They used to apparently have one issue every 2 months. Then they brought it down to 1 month, then I think fortnight or something. Then they started A, B, C, D and all that. And more papers getting published. So this was something that a physics professor told me. So he said, there is this estimation they made that by the year 2100, if you stack all the physical reviews one on top of the other, the height of the stack will increase faster than the speed of light. But the counter to that is, don't worry, it doesn't violate any laws of relativity because it carries no useful information. Okay, next. So there are also collective human limitations which are even more serious. Functioning as a group instead of as individuals, we have very inefficient methods of communicating knowledge to each other. We have to talk, we have to write, we have to show figures. These things will always limit the way we work as a group. If you are one person who knows everything, you are far more powerful, far more capable of applying your knowledge than we can ever be as a group of separate, disconnected specialists. And that's the point I wanted to make. Another problem, large populations have never solved the problem of reconciling individual priorities with collective good. So we have no collective identity. As the saying goes, we are individually helpless and collectively irresponsible. Now it's again all about human beings, and it's about how this is going to affect or challenge the sustainability of human progress. Because all the science and technology isn't going to make us behave better. We are going to behave the same way. As I have said here, we seem to be, war seems to be the popular final way to settle many questions. Our inability to avoid or minimize aggression and to simply compromise and get along has not improved one bit since the Dark Ages. It's not likely to change in future. We will remain a squabbling species. Gain is the decider. There is no a priori commitment to non-aggression. The argument meant to apply to relationships between nations. Finally, this economic compulsions and the finite universe. There's also the question of distribution of whatever goods. Oh, sorry. Yeah. You miss it every alternate slide. Okay. So what we are saying is, whatever goods we do have, whatever benefits of science, technology, etc., are distributed very unevenly within the population. And that gives rise to a lot of social tension. Now, within self-governed communities, there is no sense of equity. Political ideologies have tried to distribute wealth and social good, and okay. The winning ideology is the rule of the currently powerful and one that defines success by the amount of growth. So as long as you believe that growth is the measure of success, and growth, since it's measured in percentages, will always be exponential growth. So you are trying to do endless growth in a finite universe. That's just not going to happen. But more importantly, there is also the problem of growth depending upon consumerism and things like that. These will force you, these will help you to grow faster and at the cost of a lot of expensive depletion of resources and things like that. So a lot of quality of life sustainability issues come because of depletion of resources, and a lot of the depletion is occurring because of excessive unnecessary consumption rather than just population rise or something like that. The economic system is responsible for that. Finally, now we have never solved the problem. Okay, we have no collective identity. Okay, this slide got missed. So finally, who will get hit first? Quite apart from the collective pressures, when water becomes scarce, different sections of society are affected very differently. In the ongoing heat wave, the middle class and above will only grumble about their higher electricity bills. The underprivileged will suffer much more. Second, it came. Okay. So you see, this is a problem. So because of this structural flaw, resource non-sustainability will hit us neither equally nor simultaneously. No sudden water outage in the bathroom tomorrow morning for us. It will come as a calibrated progression of deprivation from the bottom up. This, of course, is already happening. Look at our villages, look at our slums. They don't have what we have. And it doesn't matter to us, we hold these nice symposia here and have lunch after that and all that. But the point is, the crunch is already there. It's been happening for a while. And thus, non-sustainability takes on a more subtle, softer form. That makes the situation more serious. The privileged will take longer to feel the pinch and will continue to make irresponsible choices. Fine. Finally, this for me is a very, very important last but one slide. One is now witness to some very alarming regressive tendencies in the general population. In the social debate of how bad modern science is, the public bases its criticism of the scientific enterprise on all the wrong reasons. We seem to be heading back towards the dark ages with post-truth and revisionism. Granted that allopathic medicine is fraught with side effects, overexploitation of resources has caused pollution, and nuclear weapons threaten our civilization. All these are the things that people throw at you when you say, science is bad for human beings, or science is bad for us. But the success or failure of modern science and our preference or non-preference for it needs to be decided after comparison with the alternatives that we have. What are the alternatives we have? I'm not saying that we don't need to change ourselves, but it is no point, the alternative certainly cannot be to relapse into non-rigorous systems of knowledge. That, I think, is something we have to agree. This is one clear step forward we have taken into a blind belief in the omniscience of our ancients, that they possessed the solution for all our current problems and they were wise and we people are stupid. This kind of regressive thinking, I think, is very, very worrisome. And it's happening all around us. There was, on YouTube, you can see one Indian Science Congress talk, which I think many of you would have seen, where you can't imagine the kind of things people said. Very worrying. For example, it has been argued that technology in the hands of the privileged has been used as a force multiplier to exacerbate social inequity. But the counter to it is that human knowledge is now also much better disseminated, and this helps the underprivileged to also fight back and aspire for greater things. Finally, okay. So what did I say? I spoke about epistemic and procedural fallibilities, irrespective of the agent. Second, agent fallibilities at the individual level, irrespective of the method. Sensory limitations, whatever, memory. Collective fallibilities as a group. Economic system, the compulsions of the economic system we have adopted, that is probably the only thing we might be able to change sometime if there is a revolution or something. Political backwardness, we are unable to reconcile collective good with individual priorities. The impact of inequity on sustainability, how it gives you a progression in deprivation rather than hits everybody equally. And finally, U-turns and intellectual decline. The withdrawal of popular support. If a large number of people, maybe in our country, decide that, science is all evil anyway, we knew all this and better than this 2000 years ago, then we've had it. This will be the last INSA symposium. Okay? Maybe they will call it the same thing, I don't know. But so this is essentially the thoughts that I put down for my presentation. Now I'm done, and questions from anybody if there are anything. Yeah.
Or is there any comment or question to him? A brief question. Yes.
Yeah. Oh, okay. Yeah, but at least there are several other incidents where it's just the lack of foresight that leads to things that happen. And as we say, you cannot think of what you cannot think of, if that makes sense. Yeah. So this leads us to this the tentative nature of everything we do. You do something and you can only say, this looks okay, but I hope I have not missed something. It's always possible to miss something. Sorry, I've taken a... Oops.
So may I now request Dr. Sabyasachi Sanyal from CDRI Lucknow, and he has been a very prolific scientist. He has been working on metabolic diseases, including type 2 diabetes, non-alcoholic fatty liver disease, and skeletal muscle atrophy. He has 2 patents so far and has published more than 75 papers with a very high citation scores. And Dr. Sanyal is also the fellow of Nasi Allahabad. He shall be speaking on the female hormone estradiol overcomes the adiponectin resistance in diabetic mice, a study deciphering the higher metabolic fitness in females. So may I request Dr. Sanyal to...
Thank you, ma'am. It's really nice meeting you after a long time. And thank you all. Thank you all. And I particularly thank Saman for this wonderful opportunity. And thanks to INSA as well. So I'll straightway start the talk because we are all hungry right now. And at a metabolically very critical point. So it's not a great time to talk about metabolism, but hope you can understand what I'm saying. So we know all about diabetes. I mean, everyone is aware. Almost 1 in 3 is at a risk of this particular disorder. And we all know about insulin and insulin resistance. This is pretty common. So I'm not going to talk much about it here. But what are the causes of diabetes? If we look at it deeply, so since from like human samples, we cannot take much. We take blood and do the analysis. And also like when you consider the clinical data, it is very important to get the clinical data. But the problem arises is once a person is diagnosed with diabetes, obviously that person is going to get treated. So the data points from when the treatment starts, how much improvement has actually taken place, no idea is there. So the thing is that we have to do some animal studies to actually get the clear picture. And like, it is not ethically really great to torture the animals, but we can still, within ethical boundaries, we can keep them untreated and observe what are the things happening. So if you look at diabetes, it's mostly because of energy imbalance that is occurring there. Diabetes or any other metabolic syndrome that includes these phenotypes. So when the caloric intake is high and the expenditure is low. So what do you mean by expenditure? One is obviously your physical activity, so how much you are burning that calorie. Second is, with age, our metabolism slows down. So the resting metabolic rate, that if that slows down, then again there is a caloric imbalance. So what happens to the excess calorie? That is deposited in specialized organs for energy storage. Those are mainly fats. So fats are pretty good organs. They are not, there's nothing bad about them. But once they cross a certain threshold level in the body percentage, some of the fats, they start behaving in a very disturbing manner. They become inflammatory. So there is another point to it, that inflammation itself can cause fat accumulation, and fat itself again contributes to inflammation. So it's a vicious cycle basically that happens when this kind of disorders take place. So what we have is chronic low-grade inflammation, and there is clinical data is clear about it. From the preclinical data, we find, because we cannot take muscle biopsies or other things very readily, so we have to depend on the preclinical data, and we find that mitochondrial dysfunction, chronic oxidative stress, they contribute to this whole scenario, this whole vicious cycle. Now about diabetes, you know about basic things about insulin, glucose, and all other things, but there's some unknown facets, or rather lesser known facets, that are equally important. I'm going to talk about them. So now the question is, so these things are happening. Now, is there a candidate biomolecule that is inside us, that is endogenously expressed, and preferably non-steroidal, that can address all these concerns? And it's not progressing, the slide. So do I have to point somewhere? Yeah. So the molecule is called adiponectin. So it's not very famous in the general public, but if you look at scientific domain, it is pretty famous. So this was discovered in 95, and there are 23,000 PubMed entries. What is most important thing about this particular protein is that this is available in the serum in the microgram level, which is pretty high when you compare it with something else like estradiol or progesterone. They are available at picograms level. So picogram, nanogram, and we have something here that is available at a very high amount. That essentially means it's probably very safe for the body. And now, when the depletion of this particular factor occurs in the blood, that has been associated with multiple lifestyle disorders. They include obesity, insulin resistance, atherosclerosis, type 2 diabetes, various cancers, fatty liver disease, kidney fibrosis, etc. So paradoxically, in some cases you see this adiponectin level is actually elevated. Now from human data we couldn't get that, but from preclinical data in mouse and other animal models, we know that that is not harmful actually. What is happening is adiponectin resistance is happening. So like in insulin resistance, the insulin receptors, they kind of become less active, or the pathway is actually compromised. In here as well, the adiponectin receptors, they become less effective. Now there are two primary receptors. There are more actually, but I'm going to talk about them. So they are highly expressed in either skeletal muscle or liver, and are involved in all these pathways that include glucose lipid metabolism, inflammation, mitochondrial biogenesis or function, and chronic oxidative stress. So it has huge therapeutic potential. But the problem is, even if it has a huge therapeutic potential, it cannot be translated. So that means, I mean, you have systemic depletion. So what do? You naturally give more adiponectin and hope for the best. But we cannot do that because it's a complex protein. So to put it into perspective, human insulin, which is given, this is 5 times the size of human insulin. And moreover, it has like complex multimerization properties. So larger scale production is not possible. So we use this form, globular adiponectin, in the latest studies. I will show you the slides. And the amount we use is roughly few grams. And if we had to purchase that from market, those few grams, it would have costed us 600 crores of Indian rupees. So 10 kilos of adiponectin at your hand is probably equivalent to Reliance's net worth. So now these are the receptors. You can hear me, right? Yeah. So these are the receptors, ADIPO-R1 or 2, and they participate in all these pathways like decreased gluconeogenesis, decreased lipid synthesis, fatty acid oxidation, improved insulin sensitivity, and all these things. So how they achieve it? Through regulating all these pathways, and the hallmarks of adiponectin signaling will come to this. These are important. Please remember these factors. AMP kinase activation, P38 activation, and AKT activation. These are all kinases involved in energy metabolism. And there is a super factor called PGC1 alpha. This is an inducible protein. Actually, you say like after exercise, you need a cold bath or cold shower that will actually improve your endurance and other capabilities. That is probably because of this factor. So this factor is goes up under cold shock and is pretty transient in nature and does a lot of important functions. There are other factors like PPR alpha, CD36. These are involved in fatty acid metabolism, and you have uncoupling proteins. These are equally important in terms that you can see like there are some lean people who have very high body temperature. That means they are dissipating heat, and that is probably occurring due to UCP. So what happens there is that your ATP production, if it is too much, so it needs to be dissipated. So the uncoupling of mitochondrial oxidative phosphorylation and electron transport chain happens, and due to uncoupling proteins, and the energy is dissipated as heat, which is again a protective mechanism for cold adaptation. And then you have glucose transporters as well. So now adiponectin resistance, if it happens due to lowering down of the adiponectin receptor expressions, it goes hand in hand with insulin resistance. And the situation that is created is ultimately a vicious cycle that causes progressive degeneration of the diabetic condition. If left untreated, it will ultimately lead to severe metabolic abnormalities across various organs and is life-threatening. So I said earlier that producing adiponectin as a therapeutic option was the non-existence. So the only probable way to address this would probably be small molecules. Now, I didn't have any idea of this receptor or its activities, but we were looking at the mechanism of action of a compound isolated from this plant. And this was identified as an osteoanabolic agent, that is bone anabolic agent. So when we tried to understand the mechanism of action of this compound, after intense exploration of more than 4 years, we found that this molecule is actually heating ADIPO-R1 and R2. So the binding studies are here, and if you overexpress ADIPO-R1, then this intensity of the effect went up. If we knocked them down or depleted the receptor expression, then the intensity went down. And we thought lottery lag gayi. So what we need to do now is do it in vivo. So naturally, anabolic, it's all fine, but adiponectin function is mostly like you can see in diabetes and other metabolic diseases. So we wanted to check the in vivo efficacy in diabetic mice. Now, these are genetically diabetic mice. So leptin receptor gene is mutated. So if you look at the diabetic mice, they are called DBDB mice. So there are 2 strains available. One is B6DBDB and one, this strain accidentally was mated with some other strain of mice, and that created a BKS-DBDB model. Now what happens in both of them, they are diabetic, obese, everything is there, but this strain is very resilient to diabetes. So after a certain age, their glycemic status almost remains the same. Insulin goes up, and the insulin, which if it goes up in normal cases would cause a lot of devastation in the body, it doesn't do anything here. So this mouse survived the normal lifespan of 18 to 20 months. But these mice, there is a progressive degeneration effect, and they die within 10 months of age. Now, we didn't have diabetic mice we had here, but the colony was not enough. We had the KSDVDB, the other one, the one with like progressive degeneration. But those mice were that time not available with us. Contacted our collaborators at Zydus Research Center and did this analysis. So after like 4 weeks of treatment, there was remarkable improvement in a lot of factors that include blood glucose, the gluconeogenesis. You can see the pancreatic beta cell health was much elevated. And if you see the NFLD was gone, a lot of these factors that include inflammatory cytokines, they all went down. So it was a brilliant effect that we saw. But now we became greedy. We wanted to file a patent. So for filing a patent, what you need is some kind of comparison with standard of care drug. So what we did is we didn't still have the animals at our disposal. We had some female animals. And normally people don't want to work with females when it comes to diabetes. The reason is that they have their estrous cycle. So there are a lot of hormonal changes and there can be a lot of deviation in things. So they prefer male to be the safe ones. But we didn't have male animals. We tried it in female animals and they worked wonderfully. So after 15 days, we saw glucose lowering effect. So we thought we didn't have the animals, so let's outsource it. So we outsourced it to a company. And there they used the same strain, they used the male mice. And we were shocked to see that there was no effect at all. So now basically this is devastating. So what happens is there, when we devise hypothesis, and that has some connection with ego. So if it fails, we get devastated, we try to bend the results to fit our hypothesis. But let me tell you, to the students particularly, is that if something does not match your hypothesis, that means a far more interesting thing is happening. So you search for it. So we started searching. Now, obviously, I told you earlier that if insulin resistance happens, insulin level goes up, adiponectin receptor level actually starts coming down at the mRNA level. We found that, well, it was coming down in both the DBDB mice here. So there was no problem. This was coming down. This is healthy mice, and these were coming down. But there was no difference between these two diabetic animal groups. But when we looked at the protein level, there was a huge difference. So these DBDB, the good DBDB, they didn't have much problem. In the skeletal muscle or in the liver, the adipo R1 and R2 levels went down. And it was like even more manifested in the membrane fractions. So we took muscle cells from these mice and we gave them globular adiponectin or our compound GTDF, and we saw whether they're responding. And as you can see, in black 6 mice, that's the normal mouse, it's working. In DBDB, that is the good DBDB, it's still working. But in this case, you can see absolutely no difference. So we are looking for what are the factors that are actually causing this post-transcriptional. So RNA is transcriptional and RNA to protein is translational event. So what is causing this post-transcriptional differences? So there were some papers about some of the factors which are regulating adiponectin receptors in cancers. We actually checked them and we found these 2 factors called PTB. This is the RNA binding protein and a microRNA, MIT221. They, in black 6 DBDB mice, the healthy one, you can see there was very little increase with age. But if you look at BKS DBDB, PTB went really high, and here MIT221, that went also really high. So now there is a connection between estrogen and MIT221, and that connection has been established in breast cancer. So the question was, the observations were these. So the question were, is there a sexually dimorphic adiponectin sensitivity in BKS DBDB mice? Is the mechanism similar to what we observed in B6 versus BKS DBDB? So we are talking about female mice because of this slide. So in female, we saw the effect. In the males, we didn't see the effect. That's why we asked those questions. Now, if you look at male versus female, what is the most obvious difference other than the phenotype? That is, presence of this hormone is CYP19A1. It's also present in male, but if you see female, it's present at a much higher level. So both male and female, they produce testosterone, and this is modified. This modifies testosterone to estradiol. So if you look at this CYP19 expression, So in the ovary or female reproductive area, it is pretty highly expressed. It is also expressed in some fat cells that include mammary fat pads and this fat pads in the thigh, that's the femoral fat pad. In male, it is expressed mainly in the abdomen, very little amount, and in some amount in the gonadal region as well. So if you consider metabolic actions of estradiol, it's anti-inflammatory and immunomodulatory. And postmenopausal women, they typically have higher risk of metabolic disease. So in postmenopausal women, obviously the estrogen goes down. Oophorectomy, that is removal of ovary in certain medical conditions, they can cause insulin resistance and diabetes. And CYP19-1 mutation in males, so it is equally important in males. Even in males, this is mutated, that causes metabolic disorders. And if you look at fat deposition and macrophage infiltration, that means the type of fat, whether it's a kind of like, neutral type of fat or inflammatory fat, that is also determined by estradiol. So there are, you can see a lot of colleagues of us even, oh, sorry, it's, so why some obese people have normal lipid profile and are disease free? It's probably because of this. The fat deposition in males are mostly abdominal and in females, they are peripheral. So what happens is that, so we took globular adiponectin and treated them to male versus female mice to see this. And if we find, so body weight, feed intake went down, body weight went down, and lean mass, they were increased in female mice, and fat mass was decreased, but nothing happened to the male mice. So we looked at glucose, and the same thing happened. In male mice, there were no change, and the female mice, there were a lot of improvement. So we looked at the adiponectin sensitivity indicators, that the pathways that are regulated by adiponectin. So if you look at this, all these factors were up on globular adiponectin treatment in female mice, but nothing happened in the male mice. So now we looked at those factors, the PTB and ME221 expression, as well as adiponectin receptor expression across different age groups in female mice, because this is a progressive disease. So in female, it was always high. In male, it went down. So if you see here, the graphs, it goes down. And if you look at PTB and ME221, they were correlated with this. So now the question was, okay, male mice have this problem, female don't have the problem. So if we inject male mice with estradiol, can it reverse the condition? So it did happen. So we did a pilot experiment. So we treated this male mice with estradiol for 14 days. And after that, we gave a dose of globular adiponectin for only 30 minutes to see the signaling. So if you see here, estradiol itself increased the AdipoR1 expression, and globular adiponectin increased the sensitivity. So we did a more detailed analysis, and we found after giving like estradiol for like about 23 days continuously, and after this period, we were giving like globular adiponectin, and all these improvements were occurring in male mice. So, and that was associated with higher expression and lower PTB and ME221 expression. So these observations were made. So the question now was, can the reverse be true? So if you remove this estradiol from female mice, will they succumb to metabolic disorders? So we did ovarectomy. We removed the ovaries for like, after removing the ovaries, we waited for 6 weeks and then treated them with estradiol for 7 days and did sacrifice. And if you see here, adiponectin receptor level went down, PPR levels went down, all these factors which are receptive to adiponectin receptor signaling, they went down. So AdipoR1 RNA as well went down to some extent, and these factors also went down. So also there were improvement in glucose tolerance in these mice, and that was that could be correlated with adiponectin receptor expression. So the question now was whether estradiol and ER directly influence, so whether this is happening because of some indirect effect or some direct effect. So for that, we needed experiments in a secluded environment, and that is in cell lines. So this cell line, MDMB231, it doesn't naturally express adiponectin receptors, estrogen receptors, and has a low level of adiponectin receptors. So when we introduce estrogen receptors in there, here and here you can see adiponectin receptor level went up and these two factors, they went down. So the thing is that this was happening there. So we wanted to check whether PTB and MID221 both are directly regulated by estradiol. Since we are addressing skeletal muscle, we wanted to check whether these are happening in the skeletal muscle itself. So I'll not come to this. So we created a situation of hyperinsulinemia. You look here, in hyperinsulinic condition, naturally adiponectin mRNA goes down, that is known. So the protein also went down. Now PTB is one factor. When differentiation in situ-sitial myotubes, myoblasts take place, PTB goes down, but it never came back when we treated them with estradiol. But MID221 level were rescued. So the thing is that what we ultimately found is that in male mice, estrogen level is pretty low. So that causes downregulation of adiponectin mRNA as well as upregulation of these factors, and they total come together to cause a downregulation of the adiponectin receptor protein. And in female mice, they are still up because E2 will actually inhibit these factors and ultimately will rescue the phenotype to a certain extent. So females are actually much more protected when it comes to this. So the icing on the cake is that when we check B6 and BKS DBDB animals, we found that if you look age-wise, B6 DBDB male mice has huge estrogen levels. So that actually protected them from all the side effects of hyperinsulinemia. And in BKS DBDB female, the bad DBDB mice in which males are really, really sick, they also had pretty high level of estradiol. So that's what, in our cases, showed that these mice, how they become adiponectin sensitive. So the inferences are females at reproductive age are metabolically fitter compared to postmenopausal women and age-matched female. And this metabolic fitness is due to the female hormone estradiol. E2 reverses hyperinsulinemia-driven suppression of adipo-R1 expression by countering insulin's effect at both transcriptional and post-transcriptional level. E2 ameliorates both insulin and adiponectin resistance. So these are the observations that we gained. And what we can suggest from this study is that, well, a lot of people, they have like progressive diabetes and they're rather treatment refractory. So what we can do after proper clinical trial is give them dietary intervention that contains phytoestrogens or actually increase estradiol levels in the body for a certain short period of time that can range from like a week to a month. And then probably the diabetic medicines would work better in them. So that's the kind of message and advice to all males here: get in touch with your feminine self, get some estrogen up, and that will help you. So thank you.
Thank you, Sanal, for a very interesting talk. I'm sorry I had to interrupt you in between because the time was less. So I think, is there any question to Dr. Sanal? So I have a question, if I may ask. Actually, you have generated different phenotypes which are depicting or which have insulin resistance.
Yes.
So all these, like where you are giving estradiol or you are also having this overactomized animals. Have you checked the adiponectin levels?
Yes, ma'am. In overactomized animal, if you go to this slide, I didn't have time to explain this.
Yeah.
But if you go to where is the overactomized animal, this is the over. Yeah, actually, oh, yeah, here. If you see adiponectin levels in the overactomized animal, they were much higher. So that's why, without even adding globular adiponectin, things were happening. Yes. So they were getting globular sensitive.
So thank you so much. And the second session comes to an end.
And again, I'm saying is that here condition, it's always a more controlled environment. You know what to expect, and from where you can like the outcomes. So that is why I put them those slides.
Have also discussed very interesting way of like how they look at in their labs in the different aspects of science. With this, I also want to thank Saman for giving me an opportunity to be here, and I request now to.
Lunch. So and we were hoping to get back at 2:20, but I think that's not going to be possible. So let's keep it 2:30, but 2:30 sharp. Okay. So guys, please come back here at 2:30. And for the students who are to speak now in the afternoon session, please load your presentations here in the site. Please come on now and load them, and then join us for lunch in the canteen. Right. So thank you. We'll just break for lunch now.